well, good morning, everyone, and welcome to this joint virtual meeting of the British Pharmacological Society and the British Toxicology Society. And the theme of this meeting is AI-driven innovation for smarter and safer drug development. So my name's Ian Coppell. I'm Professor of Pharmacology and Toxicology based at the University of Liverpool in the UK. I'm the current chair of the British Toxicology Society's scientific subcommittee, and I've also chaired the organising committee for today's meeting. So it's a real pleasure to have you all with us today for what promises to be a stimulating and forward-thinking online meeting. As I said, this is a joint meeting of the two societies, the BPS and the BTS. And these are the biggest societies for pharmacologists and toxicologists in the UK. And we're welcoming attendees that are members of the two societies, but also non-members who we hope through this meeting will get a feel for some of the benefits of joining the societies and consider doing so afterwards. If you're interested in joining the societies, there'll be details of how to do that shown during the various breaks. So this meeting brings together stakeholders from a range of sectors to explore how artificial intelligence is transforming the entire drug development pipeline from discovery all the way through to clinical application and regulatory assessment. And over the course of today, we'll hear from an impressive lineup of speakers who are at the forefront of applying AI and machine learning to real-world challenges in pharmacology, toxicology, and related disciplines. So we've structured the event to not only highlight opportunities, but also to discuss some challenges around the use of AI in drug development. We'll begin with foundational insights into how AI and machine learning are currently being used to accelerate drug discovery, followed by some perspectives on enhancing drug development. And later we'll delve into how clinical and regulatory assessment can benefit from novel AI-driven strategies to improve both efficacy evaluations, safety assessments, and ultimately approvals for novel drugs and medicines. So we're also excited to hear from our invited oral communication presenters who've been selected by the organising committee following a review of the abstracts that we received late last year. You can find the programme for today online. It does include a few last-minute minor changes, but just to highlight that in each of the main sessions, we'll hear from two expert speakers, and this will be followed by some time to ask questions to either speaker or both if there's an opportunity for a wider discussion. So please use the Q&A feature during or after a presentation to ask your question, and the chair will address these to the relevant speaker during the dedicated discussion part of the session. And please indicate which speaker your question is directed to if it's not clear. There's a chat facility too, but we ask that you only really use this for general comments and not questions, as the session chairs will only be monitoring the Q&A for questions. So please use that for your questions. So thank you for joining us. Thank you in advance to all of our speakers for sharing your expertise with us. I hope you find the meeting informative and inspiring as we collectively consider the next steps in AI-enabled drug development. So with that, I think we can move on to our first speaker, who's Dr. Peter Cox from Isomorphic Labs. So Peter obtained a PhD in molecular virology from the University of Glasgow, and then he undertook postdoctoral training in neuroscience at INSERM in Paris and the University of Cambridge. Peter then began his industry career at Pfizer, where he led drug discovery projects through to IND preparation in the area of chronic pain therapy. And in 2014, Peter joined Benevolent AI, where he was instrumental in building and expanding a team of drug discovery scientists and establishing a robust portfolio of projects. And Peter is currently head of translational science at Isomorphic Labs, which is responsible for the biology strategy and in vitro validation of the company's AI-led drug design projects. So Peter's going to be giving us an introduction to AI and machine learning in drug development, which will lead on nicely to today's focus sessions. And as I said, if you think of any questions during Peter's presentation, please use the Q&A feature, and we'll try to address as many of those as we can afterwards before Peter has to leave. So, Peter, thank you for kicking us off. Over to you.
Excellent. Can you hear me okay? Yeah.
Yeah, that's great.
Can you see the slide deck as well?
We can. That's perfect.
Excellent. Just bear with me. And you can see my laser pointer, I assume, as well.
Yeah, we can. That's great.
Brilliant. Okay, so firstly, I'd like to thank the organisers, and in particular Professor Koppel, for the invitation to kick off this event in introducing artificial intelligence and machine learning in drug discovery and development. These are the topics I hope to cover today, with an unashamedly large focus on what we are doing at Isomorphic Labs as an exemplar of what is happening in the AI ML space to enhance drug design. Along the way, I'll also touch on some of the translational aspects as well, and finish off with a brief discussion of what AI in the future might mean for us as drug safety scientists, toxicologists, pharmacologists. As Professor Koppel has outlined, I started my career in drug discovery in large pharma at Pfizer before moving to Benevolent AI, one of a number of companies that were the first wave of AI-led biotech. And 2 years ago, I joined Isomorphic Labs, who are focusing on developing and capitalizing on increasingly powerful AI ML-based drug design systems. And if I may, I believe I've had a front row seat over this past 10 years to the emergence of AI-led drug discovery. And I'd be the first to admit, as with most new technologies, it has had its teething problems, and there is certainly much hype surrounding AI in drug discovery, and you may accuse me a little bit of that today. I believe that second generation companies like Isomorphic Labs are really beginning to address the complexities of biology and the data required to effectively train predictive AI models with the potential to impact end-to-end drug discovery. And I think it's a technology that we should be embracing and developing as a significant part of our arsenal to understand complex biology and its implication for drug design and drug safety. So there are 4 key bottlenecks of drug discovery that AI-first companies like Isomorphic Labs, and indeed academic labs, are addressing. The first bottleneck is understanding disease and target biology with the aim to identify and validate the right targets. It doesn't matter how good, how efficient an AI-led drug discovery program might be. If the selected target is incorrect from the get-go, it is obviously doomed to failure. The second bottleneck is being able to identify the best ligands for a given target from vast chemical and biological space. And the third bottleneck is to develop those ligands into high quality clinical candidates that are unlikely to be toxic and have good ADME properties, and will allow further development of those ligands into the clinic. The fourth bottleneck is being able to do this in a generalizable way across many different target classes, diseases, and drug modalities. And I put it to you that solving one or a number of these bottlenecks has the potential to accelerate the drug discovery process. We can get to the clinic faster, hopefully with more successes, and if not, we learn from those mistakes, hopefully quicker. AI is already facilitating an understanding of biology at many different scales and complexities. Isomorphic Labs are developing machine learning models that provide atomic level resolution of biomolecules. In addition, we are building a drug design engine to efficiently design drugs for any target and disease by unlocking significant bottlenecks in multi-parameter and multimodal drug design, including predicting toxicity. And we believe that the work that we are doing to understand biomolecular interactions at scale will also enhance the use of AI in the analysis of complex multimodal data to yield new targets, biomarkers, and a different, more nuanced understanding of disease. So what is AI/ML? Artificial intelligence is essentially computationally achieving or surpassing human intelligence, and it's underpinned by machine learning computer algorithms that are trained on large data sets to learn to perform complex tasks or predictions at scale and speed. I'm going to give you a very basic explanation of AI/ML, but if you would like more detail, I'd highly recommend this review from Seal et al. In 2025 that discusses various types of AI/ML models in more detail and the considerations for their use, particularly in predictive toxicology. So why do we need machine learning? Some tasks are relatively straightforward, like baking, maybe, or calculus, again, maybe, in that you can follow a specific set of instructions to achieve an outcome. Other tasks are learned by example or trial and error. A case in point is protein structure prediction. Some proteins are highly structured, whereas others are more disordered or dynamic. So machine learning would need to learn to predict protein structure by being exposed to many different diverse examples of protein structure. And this is essentially how the protein structural prediction model AlphaFold was trained. Machine learning is about teaching computers to solve complex tasks through exposure to many examples. And there are three core components to machine learning. The first is data, and as an experimental biologist, I think this is the absolutely most critical aspect of machine learning. We need and we continue to need high quality data sets to train, test, and evaluate our models. The models consist of many different layers, and underpinning those layers are various learning algorithms. Machine learning models are trained on real experimental data and then used to make predictions, initially with query test data. The learning algorithms compare the predictions against the test experimental data and calculate the difference between the test data and the predictions to compute what is known as a loss function. The learning algorithm will then iteratively learn which parameters within the machine learning model to adjust to minimize loss, to improve model predictions. And this is known as gradient descent. It's a process that I'm not going to go into any detail. I don't really understand it in great detail myself. So machine learning models are evaluated by splitting data sets, experimental data sets, into test and training sets. One key question is how to make these splits. Your train test split should accurately represent the difference between your training data and the query data in practice. Common splits are splitting the data by time or random splits. And I'll show you a few examples where we have tested our systems using published data that emerged after the cutoff date for the training of our models. As I've said, the models are trained using a process called gradient descent, and then they are evaluated using evaluation metrics on the test set of experimental data. And the evaluation metrics that are used really depends on the use case. And for example, in protein structure prediction, there are a variety of statistical endpoints, such as root mean square deviation, which measures the predicted versus observed values. That would be the predicted protein structure versus ground truth X-ray crystallography data, or the local distance difference test, which evaluates local distance differences between all atoms in a model, including evaluation of stereochemical plausibility. In terms of AI classification models, recall and precision are used, and I'll explain this in the context of a typical AI drug discovery model that would be used, let's say, to predict the binding affinity of small molecules for a given target of interest. So in this instance, recall is the fraction of true binders identified, and precision is the fraction of correctly predicted true binders to the target of interest. So it's really important that for both training and testing a model, in this instance, a binding affinity model, that we would have quite a diverse set of both true positive and negative data. That's absolutely essential for training. In terms of generative AI, unlike traditional AI that categorizes data, generative AI produces new material based on a user's prompts or inputs by predicting and synthesizing the most statistically likely responses. Generative AI with diffusion models can create realistic new data, such as images or text, by first adding random noise. You can see this here, this image of the dog. It becomes much more noisier And then the models are trained to actually take images like this and denoise them in a stepwise process. And they can ultimately then take new prompts and generate new structured data from the random noise. More and more, we see foundation models coming into place, and these are machine learning models that are trained to perform multiple diverse tasks. And because of that, they're trained on very different diverse data types and can be adapted or fine-tuned in order to perform these specialized tasks, which is really a nice segue into some of the foundation models in drug discovery that I would like to talk about, namely AlphaFold 3 and Isomorphic Labs drug design engine. Atomic level resolution of biomolecules has been initially facilitated by AlphaFold 3, developed through a collaboration between Isomorphic Labs and Google DeepMind. AlphaFold 3 really represented a step change compared to the state-of-the-art methods to model protein-protein interactions, antibody-antigen interactions, and complex biomolecular interactions, including those between proteins and nucleic acids. And the two examples in this slides are, on the left-hand side, the large coronavirus spike glycoprotein in complex with neutralizing nanobodies, and on the right-hand side, an RNA polymerase enzyme in complex with primers and a nucleotide. Both of these complex biomolecular structures were predicted with a high degree of accuracy by AlphaFold 3, despite the fact that it had never seen either of these structures in its training set. And you can actually see this more clearly here in the magnification, let's say on the right-hand side. So the ground truth X-ray crystallography data for this biomolecular structure is represented by the gray. So, and then you can see in these somewhat lowered colors the AlphaFold 3 predictions for the protein, the nucleotide, and the primers. And you can see there's quite a significant degree of overlap, suggesting that AlphaFold 3 can accurately predict complex biomolecular structures at high resolution, despite, as I said, having never been exposed to these types of data in training. We can compare AlphaFold 3, we can quantify how it performs in comparison to other state-of-the-art methods. I'm not going to labor this point to any extent other than to say that when it first emerged, it was quite clear that AlphaFold 3 tended to perform better than state-of-the-art methods on protein-protein predictions or protein nucleic acid predictions. But as you can see from this slide and the previous slide, there is some room for improvement here in terms of AlphaFold 3's predictive performance in the context of both in a variety of different contexts in terms of predicting biomolecular structure. So how are we building on these significant achievements for drug design at Isomorphic Labs? We are evolving structural prediction capabilities beyond what was previously achieved with AlphaFold 3 and combining it with a whole suite of additional predictive models of relevance to drug discovery. And I think the most important aspect of what we are doing is that we're placing this powerful drug design engine in the hands of our drug designers who are using this in our portfolio of targets in real time, and they're feeding that information back into these predictive models to optimize and refine those models further within our drug design engine. We are continually evolving our structural prediction capabilities beyond what is currently achievable with AlphaFold 3 across all types of biomolecular predictions. And in this specific example on the right-hand side, you have the ground truth structure. So this is X-ray crystallography data showing a small fragment-like molecule in complex with a DNA glycosylase enzyme. Our latest ISO structural prediction models can predict the location and the pose of the fragment quite correctly, whereas AlphaFold 3 gets the location of the fragment correct, but the actual pose itself isn't quite as it should be in comparison to the ground truth structure. And this is reflected in the fact that the RMSD score for the latest ISO structural model is lower, therefore much better, more accurate than the AlphaFold 3 MSD value. Isomorphic labs are developing models that can predict and rank the binding affinity of large virtual compound libraries for any given target. And what we do here is we take a target protein sequence of interest together with a virtual library of SMILE strings representing all the molecules in that library. And we input this information into a neural network-based model, which can then predict and rank the predicted binding affinity of these molecules for any target of interest. And we have identified hits for every project using this type of approach. Even more exciting is our AI-directed de novo design approach, in which we use the same binding affinity algorithms, but include other design criteria such as predicted ADME properties or predicted phys-chem properties to generate entirely new chemical matter. So this differs from the previous approach in that it's not dependent on the input of a virtual library. It's not a virtual screening approach. It is actually generating new chemistry de novo. And both of these methods reduce our need for experimental screening. They have provided consistently good hit rates across our platform. And certainly the de novo generative design system is also leading to the identification of novel binding sites on some of our targets of interest. The de novo generative AI model is being applied to drug discovery projects at ISO with some compelling results. Importantly, this powerful AI model has facilitated the identification of active compounds for 7 targets within our portfolio, with a relatively small number of compounds made and tested by the project teams. And the model is also potentially generalizable, given that the 7 targets, which unfortunately are anonymized here, so you'll have to take my word for it, these represent different target classes, which suggests that this model is actually generalizable across a number of different target classes. So the more confidence we can have in these rapid in silico predictions, the more we can move away from long traditional design, make, test, analyze cycles to faster, more accurate in silico based cycles in terms of expediting the hit identification and lead optimization, candidate selection aspects of drug discovery. So moving away from some of the traditional small molecule examples that I've outlined in the previous slides, I want to talk a little bit about alternative modalities. I mentioned briefly the performance of AlphaFold 3 in terms of predicting the structure of therapeutic antibodies and complex with antigens, which is a modality that Isomorphic Labs and many other companies are now exploring. And here is some early work we did on in silico prediction of nanobody sequences for 4 different target proteins. So we were trying to get our platform to ultimately predict nanobody sequences that could bind to these different target proteins. And with very little refinement of our platform, we achieved a 5 to 11% hit rate for these 4 different targets and are continuing to improve on that. Moving on to another modality, molecular glues are certainly gaining a lot of focus, not least because of the work of Monte Rosa Therapeutics and Relay Therapeutics in bringing this particular modality into the clinic with the targets that they are looking at. So molecular glues are essentially small molecules that bring in effector proteins into close proximity with target proteins, resulting in a number of different effects, potentially depending on how the interaction is facilitated. It can result in the degradation of the target protein, it could inhibit the protein function of the target, or it could sequester the target to a compartment, thereby rendering it non-functional. We are building a platform of rational design for molecular glues. Our platform is able to identify novel effector target protein pairs that it has not previously seen. The 2024 publication described SPSB3 as a novel effector protein that drives the degradation of cyclic GMP-AMP synthase, an important innate immune system target on the STING pathway. Now, I'm going to stress here, this is an example of a time split in that this publication emerged after the training cutoff date for the ISO platform. However, the ISO platform was able to identify SPSB3 from over 1,000 potential effector proteins as the protein, indicated by the arrow here, with the lowest predicted protein-protein interface error with C-gas. And in addition, our platform was able to accurately predict the structure of this PPI. And I'll stress again, it had never seen this structure. In fact, SPSB3, for one reason or another, was not actually present in our training data at all. So we believe that our ability to look for novel effectors across the proteome is an exciting and novel source of gluable biological systems. And we are using some of the de novo design approaches that I described in the previous section to identify molecular glues for novel target effector pairs. So our platform can address traditional and also emergent drug modalities, which could potentially be applied to challenging targets. So how does it perform with challenging targets? Transcription factors, as most of you know, are notoriously difficult to drug. Everything about a transcription factor presents a challenge for the isomorphic platform. But I want to particularly highlight the fact that transcription factors are intrinsically disordered structurally, and there's very little small molecule training data available for transcription factors. In fact, there are only 3 unique transcription factor uniprot IDs with some small molecule data within ISO's training data set. So there's a real paucity of data here, and they're structurally very complex proteins. Again, a literature study that emerged post the training cutoff date for our platform was used to test if the conclusions from that publication could be modeled in silico in any way by the ISO platform. The publication focused on the transcription factor FOXA1, which is a pioneer transcription factor important for liver development and associated with breast and prostate cancer. It's a complex, highly disordered protein. You can see here on the right-hand side, the AlphaFold 2 model for FOXA1. You see large areas of unstructured protein interspersed by smaller areas with defined structure. In fact, there is only one X-ray crystallography data in the protein database for FOXA1, which is the DNA recognition sequence, protein sequence of the transcription factor in complex with the DNA recognition sequence. So, as I said, publication emerged from the Cravat and Herb Labs on FOXA1, which provided some experimental ground truths that we could use to test the ISO platform. It provided DNA recognition sequences and negative control DNA decoy sequences. And really interestingly, they also had identified a covalent small molecule probe with a characterized binding site at cysteine 258 in the FOXA1 transcription factor. Our platform was able to correctly fold a high confidence structure of FOXA1, but only in the presence of the cognate DNA recognition sequence. With the negative control DNA decoy sequences, we had a very high predicted error rate in comparison to the error rate, which was quite low for the protein in complex with its recognition sequence. We next asked, could the platform identify cysteine 258 as the binding location for the covalent probe? We did not provide the model with any information other than the FOXA1 protein sequence, the DNA recognition sequence, and the SMILE string for the covalent probe. We did not bias the model in any way towards cysteine 258. So by folding this triad many times and scoring the output, our platform converged on a high confidence solution, which really placed the electrophytic probe in close proximity, as you can see here on the y-axis, which measures the distance to cysteine 258. The pose predicted by our platform of the covalent probe in complex with FOXA1 was consistent with the possibility of the formation of a covalent bond between the acrylamide moiety on the warhead of the probe with the thiol residue on cysteine 528. And again, I stress that no structural data was provided in the publication. And this is actually, by any measure, is not a straightforward prediction, because this region is missing from the ground truth structure, and it is largely disordered in the APO AlphaFold 2 model. So this is suggests that we can actually address some challenging targets in terms of structural predictions. Beyond the publication, we wanted to check our ability to identify relevant protein-protein interfaces for a highly disordered protein such as FOXA1. And we did this by using the platform to in silico screen a set of known binders to FOXA1 derived from the intact protein database of PPIs and compare it to multiple negative controls. And with this approach, we could confidently predict these protein-protein interactions from the intact database, whereas the negative controls, these were proteins that were randomly selected for the proteome or represented either secreted or membrane proteins, which were considered to unlikely to form an interaction with FOXA1. And as I said, you can see that the known binders basically had a much lower predicted error rate than the negative control proteins. I will stress that the known binders, that the PPIs, none of these, again, were in our training set for our model. So it's really not beyond the realms of speculation that we could potentially use this type of predictions to develop gluable systems for difficult to drug targets like transcription factors. I know I've gone quite quickly through this, but I hope I've given you a sense from this section of Isomorphic Lab's mission to remove some of the drug design bottlenecks, in particular in the hit ID, lead optimization, and candidate selection space, and also to develop generalizable multimodal drug design approaches, which will hopefully expedite the drug discovery process. However, AI has been used to bridge the gap between preclinical and clinical sciences. And in this slide here, I highlight 5 relatively recent publications to make a few points. AI can lead to a more nuanced understanding of disease, and a collaboration between Novartis and Oxford University used AI to show that multiple sclerosis is not just 2 subtypes, but rather a continuum of 4 different disease subtypes. Combined analysis of clinical endpoints with omics data will lead to enhanced target discovery and target safety predictions. And Google published a paper combining clinical cardiovascular with genetic readouts to really enhance genetic discovery and risk prediction. However, in many instances, access to relevant clinical data sets may be limited. So it's important that targets can be inferred from relatively sparse data sets. Work from the Broad Institute demonstrated that it was possible to predict cardiomyopathy targets from sparse single-cell transcriptomic data sets using an unsupervised machine learning model trained on a large corpus of unrelated single-cell transcriptomic data. Modeling disease and toxicity in vitro will require more complex models and endpoints provided by companies like Recursion, for example, who are using increasingly more complex AI-led cell imaging in conjunction with omics data sets to really understand or model disease in vitro and also look at toxicity from that perspective. But it's also important that these data sets can be visualized, explored, and interpreted by us as scientists to yield new insights. And this was nicely illustrated by knowledge graph network biology-based approaches of companies like Benevolent AI and the exciting work that they did during the pandemic that led to the discovery of baricitinib for the treatment of COVID. I think underpinning these advancements will be an understanding of how biomolecules interact and function to further enrich and explain AI-led scientific insights such as these that I've mentioned. And this understanding of how biomolecules interact and function at scale is really a key area of focus for Isomorphic Labs.
Peter, we're a bit over time now, so yeah, you'll be able to
Yeah, what I'll do, I'll just briefly say, in terms of clinical trials, obviously there's vast amounts of data out there that we could potentially make safety and efficacy predictions with. We could also design more cost-effective trials using virtual trials and simulations. I was going to cover a little bit about AI and toxicology. I'm not actually going to go into that in the interest of time. I do think as a community, we need to be embracing the AI revolution. We should be generating high quality data sets, which will be needed to train and test, especially in the era of drug discovery involving more challenging targets, more sophisticated drug modalities, to develop predictive mechanistic toxicity models, and to underpin more complex treatment regimes. AI can make us more efficient. You'll hear more about large language models today, but these can only improve if we point them in the right direction, ask the right questions of them, and critically appraise their outputs and sources. And if I may give one final piece of advice, particularly to those that are at the beginning of their careers in the audience, then if you really want to be part of this AI revolution, do all of the above, but also really learn how to interact and communicate with AI machine learning experts to design what I think is the future of drug discovery. And with that, I'd really like to acknowledge my many colleagues at Isomorphic Labs, some 300 experts from diverse disciplines that are really pushing the boundaries of drug discovery and development with AI first approaches. And thank you all for your time and attention, and I hope you really enjoyed today's conference. Thank you. Sorry.
All right, great. Yeah, no worries. Thank you, Peter. That was a really nice introduction, really sets the scene nicely for the rest of the meeting. There is one question that was in the chat that we could maybe spend a minute addressing before we move on, but there's a question about your de novo prediction model that you talked about. How good is the comparison of the hit rate from your de novo predictive model versus actually confirming how the chemistry behaves experimentally in your systems? Have you looked at the correlation there?
Yeah, absolutely. We verify, we experimentally test all, or quite a significant number of the molecules that we generate using our de novo design approach. And we use both binding and functional assays to do that. And we do see, as I said, quite a high hit rate, even by testing a relatively small amount of molecules in those experimental assays.
Okay. All right. Fantastic. All right. Well, thanks again, Peter. I think you need to leave now, right? So, yeah. Thank you for your...
Enjoy the rest of the conference. Thank you.
Great. Thanks a lot. Okay, I can now hand over to Alex, who's going to be chairing the next session on the role of AI in drug discovery. So, Alex, over to you.
Thanks. Thanks, Dr. Cox, for his great overview and introduction to the subject in AI and machine learning. And good morning, everyone, and welcome to our first session of the day, where we're going to continue to sort of zoom in and start at the start of drug development and focus on AI in drug discovery. So this is a stage where there's, as we've heard, there's a large number of options and a large amount of chemical and biological space open to the scientists that they need to navigate. And this is where we think that AI can help a great deal here as a tool for the scientists to make their decisions and make those choices less overwhelming. So my name's Alex Cayley, and I'm one of the coordinators of the Computational in Silico Toxicology Speciality section at the BTS, and I'll be chairing the session. But I'm glad to say that I'm also joined by two experts in this field, in Professor Charlotte Dean and Dr. Olivier Becquignon, who will be sharing some of their ideas and work in their presentations. So after the presentations, as we've heard, we'll have 15 minutes at the end of this session for questions and a panel discussion. So please put your questions, comments, ideas for discussion into the Q&A section, and I'll filter through those and we can come back to those at the end of the talks. So without further ado, I'd like to introduce our first speaker in this session. This is Professor Charlotte Dean, MBE. So Charlotte is a professor in the Department of Statistics at the University of Oxford, Executive Chair of the Engineering and Physical Sciences Research Council and co-founder of Dalton TX. During the COVID-19 pandemic, she served on SAGE, the UK government's scientific advisory group for emergencies, and acted as UK Research and Innovation's COVID-19 Response Director. In 2025, Charlotte was elected as a Fellow of the International Society for Computational Biology, and at Oxford, Charlotte leads the Oxford Protein Informatics Group, who work on diverse problems across immunoinformatics, protein structure, and small molecule drug discovery using statistics, AI, and computation to generate biological and medical insight. Charlotte's research focuses on development of novel algorithms, tools, and databases which are openly available to the community. They're widely used in both academia and industry and are embedded in pharmaceutical drug discovery pipelines. She's also a member of several advisory bodies and has consulted extensively with industry, having also established a consulting arm within her research group as a way of promoting industrial interaction and use of the group's software tools. Charlotte is part of the team leading OpenBind, which is an £8 million government-backed consortium aiming to create the world's largest open data set of drug protein interactions to accelerate AI-driven drug discovery. And she also serves as one of the 5 experts advising the UK's government's new AI for Science strategy, which aims to boost AI adoption across research and accelerate scientific discovery. So welcome, Charlotte. And I think there's been a slight change to your talk title, which is going to be AI in small molecule drug discovery. Over to you.
So I'm hoping the magic screen share has worked. Just going to take that as a yes because nobody's screaming. That's all right. So good morning, everybody. I'm going to try today to tell you 2 short stories about AI, kind of its use in drug discovery and in chemistry. And one shows the absolute power of it, and the other one shows some of the pitfalls we should think about when we're trying to do this kind of work and how we should think about doing it. So the first story starts with large language models because everybody loves large language models at the moment. Transformers have completely changed the way people think about using AI. They've kind of run into the world so fast. You know, they're being used everywhere. If you run a Google search now, it's using a large language model to produce some results up the top to give you a different description of your search parameters. They're being used in the way we interpret language, natural language. They're being used to make images. They're everywhere. And they're clearly very, very powerful. So we asked what I thought was a question that they shouldn't really be able to do. And the question was, can a large language model, not trained, don't do anything special with it, take them exactly off the shelf as they have been written by these companies, is it useful to do chemistry with? So there's a lot you could do to one of these to make them really good at this, and loads of people will talk about doing that. But just start with, how good are these models already? How useful are they to us? And in order to test that, we wrote something which we call ChemIQ. And what we wanted was a benchmark to do the testing that was algorithmically generated and that wasn't a multiple choice benchmark. It was really interesting when we looked at this. Almost everybody who's testing these models to see how well they do is doing multiple choice with them. And when I'm doing a new piece of chemistry, it's not a multiple choice question, it is an open question. So we needed open questions, and we needed to algorithmically generate these questions so we can update the questions whenever we like, because we have to stay ahead of the training data of the models to be sure they're not just telling us something they have seen before. So what do large language models do? Well, it's a fairly obvious answer to this. Large language models generate text. So if you give them a prompt, like what's the main difference between cats and dogs, this is actually one of the things you can get them to say, which I quite like, which is cats bark louder. It makes no sense, but this is what they are doing. So you can prompt them with text and they generate text as an answer to that. But large language models used to struggle with problem solving. So if you go back 12 months, if you'd asked a large language model, if you have 3 apples and eat one, how many apples are left, you would literally get the answer. Most likely answer you get would be 4. because they were unable to do the process of doing 3 minus 1 as a very simple type of problem. Now, this changed as we started to have chain of thought in large language models. At this point, it improved problem solving. So if you ask the same question, if you have 3 apples and eat 1, how many apples are left? It actually went through a process of, and I'm going to call it thinking, but it's not really thinking. It's a use of tokens inside the model to be able to do this. So the answer became, you start with 3 apples, you eat 1 apple, that means you've taken 1 away from 3, that means you have 2 apples left. So chain of thought improved the process of problem solving. So starts to be, well, maybe they can be useful in the realm of answering chemical questions. And then we got to large reasoning models, which is where the models are now. So here, what you have are extra tokens that are running inside the model. So the aim is they think before answering. One of the weird things about this is you can actually get it to kind of give you out the text of what its thoughts are. Now, to be careful here, it's not really its thoughts, and it's another model being run over the large language model to turn that inside latent space into text. So it's over the top to claim this is what it's thinking, but it gives you an idea about how the tokens are being used. So if you ask these large reasoning models these questions, if you have 3 apples and eat 1, how many apples are left? It thinks. And this is actually what it produces internally. I start with 3 apples, then I eat 1 apple. Eating means I consume it, so it should be gone. Therefore, I subtract 1 apple I ate from the 3 I had. So 3 minus 1 equals 2. That means I have 2 apples left. 2. So given we are at this state with these models, and this is a really, I mean, I've used a very simple question here, but given we're at this state, does that mean that these models can reason and think across the kinds of spaces we are interested in? And can we just use them out of the box to be able to do really useful things? So we put together ChemIQ to do that test. We're going to test 3 key competencies, and they kind of run from things which should be straightforward and certainly are straightforward to a human, to things that actually are really quite difficult even for a human. So molecular structure, carbon counting, ring counting, atom mapping. We can do that. Yeah, a human can do that, no problem. You can write a piece of code that can do that really straightforwardly. You do not need a large language model or a large reasoning model to do that. An interesting question, which is the language of chemistry, how much of that is in regular text? So can you just convert a smile string to IUPAC? So can you take what would be a standard kind of computational representation of a molecule, a smile string, to what would be our word description of it, IUPAC? Previously, models really couldn't do this. And then we go through to reasoning questions, through to NMR elucidation. Can you, if I give you the NMR spectra, tell me the actual structure of the molecule? Not running any code. So this is not using agentic AI, moving to that. You could do that, but just can these models just do this? And that for a complex molecule, even with software, a human might take an hour or so to get that right. So can these models be useful with these problems? And the answer to this question is yes. Really, really, yes. So if we move from the non-reasoning models, I'm showing you the OpenAI models here, but we did it with all the different large language models, and I'll show you more results along the way. But GPT-4 at the beginning there is just a standard large language model, and it really didn't get many of the questions right. And you can see that in grey on the left-hand side of the graph, or you can see it in grey in terms of the types of questions it got right in the actual kind of question set we had. By the time we moved to O3 Mini, which is a large reasoning model, we're starting to get quite a lot of things right. So I've done nothing. And these models are capable of answering correctly. What that means is if you wanted to do NMR elucidation, you could just throw it into a standard large language model. And quite a lot of the time, it will give you something very useful as a start point or even the correct answer. This is to give you an example of this. This is a 25 atom example where we just asked it to write the smile string, and we literally gave it what was the formula, and that's the peaks. And it gave the correct answer, which is on the right. And that is a hard chemical reasoning problem. And what's even more interesting is if you take that, the reasoning process resembles a human. So you can see here, it goes through the process of going the 1H NMR around 7 to 8 ppm. I love the fact that they've changed the language of the way when they write the code to do this. There's no way a computer is thinking screaming out for benzene rings, but they use this as a way of describing to us what the tokens are doing inside. But this is how it generates that structure. So it is doing it in a way that a human would do it, where we would say, well, those must be benzene rings. Can we assign those protons? How can we do this? How can we build each piece? Having the benchmark also allowed us to kind of do the next stage up as more and more models came out. So we've still got GPT-4 on there, where its success rate was very low, but now we can go all the way through to when GPT-5 came out, and you can start to see here real progress in being able to do a lot of these problems. So there's lots of these things these models can just do for you now, but they still can't do it perfectly. But they're an offering and a start point for being able to get chemistry directly out of a large language model as it stands. And what that means is, in the very near future, you can imagine the situation that you fine-tune these without much chemistry, but a small amount, or you allow them to run code in the way you would with agentic AI, and you'll start to see these models be able to answer lots of these questions as a standard part of what they do. So it will become many of these things that we think currently take some reasoning, working with a piece of code, building through, will now be able to be automated entirely with a large language model of this type. So my second story is the less cheerful story. So I've told you the happy story, because it's always good to start with the happiness. I'm now going to talk about something which is kind of the flaw that sits inside thinking about these models and how they work. And this is about docking. So docking is a problem that everyone here, I'm sure, is aware of. I want to work out how this small molecule binds to this protein. First question, does it bind? And then second question, what shape does it take within that? And there were a lot of papers that came out kind of 2023 time, and they weren't really papers. They were kind of descriptions of work going, artificial intelligence is better at docking than any of the traditional techniques. And I did the classic supervisor thing at this point. I said to my student, I went, all right, go and read all these papers and all these white papers that have come out, work out exactly what all of them are doing, and then come back to me, we'll talk about it, and we'll work out how we build something on top of all of this. Yep. So off he toddles, comes back, we chat during the week, but he comes back properly for next meeting the following week, and he goes, I don't think this is quite right. And I'm classic supervisor. I go, come on, you've got to really get your head around this, go and try them again, work out how they're running, we go through it all. And he comes back the following week and goes, no, really, I don't think this is quite right. So the question we were asking was, is the ability of these artificial intelligence methods better at docking than traditional techniques? Okay, so can I put that drug into that receptor more accurately than other methods could? And the next slide is kind of the demo of lots of the issues that are occurring when people use artificial intelligence for these things. So across the bottom is the crystal structure of the molecule. I haven't bothered to put the protein on here because it just makes life worse and more complex to look at. Across the top is the RMSD of the predictions I'm showing you in the middle. Now, I'm hoping that everybody on this call is wincing slightly or moving slightly in their seat or feeling a small amount of pain. My favourite one of these, favourite being an odd word to use at this point, is the second one across, where the model has predicted something with two rings that run through one another. It gets an RMSD of 1 angstrom, but every single bond within that molecule is a length which is physically impossible. And what we discovered, just by starting to look, was these methods had been told to generate really good RMSDs. So they did. But even though they'd only ever been shown physically plausible things, they weren't told they had to keep it physically plausible, and they didn't know what physically plausible meant, so they don't. So we created a very simple test set of tests to have a look at this. So we're going to test a set of methods with two data sets. The first one I'm just going to call the easy data set. That's the Aztecs diverse set. Now that is probably stuff that's in the training data of these models. So if they get that wrong, they're really not very good at their job, because they have definitely seen this when they were trained. The second, I'm just going to call the difficult data set. It's not actually that hard. And I can split that into 3 types. So this is data that was released after the training point of these models, but I'm going to show you things which are sort of high sequence identity to stuff we think they've seen, medium and low. So low is yellow, orange is sort of medium, and dark orange is very similar to what they've seen before. I'm going to test a set of methods. I'm going to test 2 traditional methods, so AutoDoc, Vena and Gold. Not necessarily the best methods there are, but just ones people use a lot, standard docking methods, and 5 AI-based methods. And these 5 were picked purely because they use very different internal kind of types of machine learning. And all I'm going to do is I created a test suite called Postbusters, and this is a set of tests to test the chemical validity of the molecules. So is it a real molecule? Then the intramolecular validity, so the kinds of things I showed you on the previous slide, it would say, no, you cannot do that with the bonds. So check the bond lengths, the bond angles, the internal energy. And then the intermolecular validity, in other words, does it smash into the protein? Is it physically impossible? And that's all I'm checking. I'm not even going to be that strict about it, okay? The Postbusters suite is all there, so you can use it to test anything, but we were relatively generous in the overlaps that were allowed or the differences in bond length. So really, we're just testing if things are really stupid, is how I would describe this. And this slide shows what you get. I'm going to focus on DiffDoc here, not for any particular reason, but it's easier to talk about one machine learning method, but the patterns are the same for all of them. So I have Gold and Vena on the far left-hand side of the slide. And you can see that for the 2 different data sets, the Aztec set and the Postbusters set, they get around 50, 60% right. And it doesn't make that much difference what you do. Yeah. And they have a small number of things that are in that hashing, which says that actually they do occasionally predict things which are physically impossible. And that's not a surprise because they have a different energy function in them from maybe the constraints I've put into Postbusters, but also sometimes they literally can't converge on a physically possible answer. And then I go to DiffDoc. So DiffDoc gets a higher percentage if I just take the number of things with low RMSD on the Aztec's data set than that which is got by the traditional methods at 72%. Yeah. So yeah, the headline claim is correct. But even if all I did was require it to be physically possible, it's already worse than traditional methods. If I go across to what is, I still wouldn't argue, a particularly hard data set, yeah, and ask it to be physically possible, it now only gets 14% of things right. So are they outperforming traditional methods? Oh no, they're not. But are they still able to do useful things? Probably yes. It's just the way we trained them and the way we tested them has not driven them to the behaviours that are important to us. The second sort of piece of results from this is probably even scarier. So this is taking that harder test set, so the Postbuster test set, but actually splitting it into those 3 bins of the training similarity to what we believe is in the training set. So the dark orange are things that are really similar to the training set, through to the light yellow, which are things which we think are really quite different. Yeah. Now, if I take Gold and Vena, it doesn't matter. That just doesn't matter. So they're about the same across all of those. So they get about half of it right. Doesn't matter which one I pick. Doesn't matter what bin I'm in. But if I go to DiffDoc, I'm just using that as the example again, if I go from where it is very similar, where if I allow it to have its physically implausible things, it's still at around 50%, so it looks very similar to traditional methods, yeah, through to this low sequence identity bin, it gets less than 1% of those right. And I should say this, I have been trying for some time now to work out if I can show that that is random performance. I can't. I don't think it is. I think it still has signal beyond random, but it's very hard to work out exactly what you would expect for random for that. But this is really important because the physical plausibility is something that people have been fixing since we first put this paper out. And that is fixable. That is the training function you put on the algorithm. You can force it to behave in a physically plausible way. It might reduce your number of kind of low RMSD things, but you can do that. You can do it in many different ways. We've done it, others have done it. But it's much harder to ask how you fix this problem with working outside your training data set. And I've put this in just in case people think, well, you've used docking, but we're not really interested in docking algorithms anymore. Everybody uses co-folding to put the ligands in the protein because that's much better. And this is work which was done by another group, but showing exactly the same pattern I have just shown you. So what they've done here is across that x-axis is similarity to the training set and your success rate of getting your co-folding correct between your ligand and your protein. And they're looking at all of the different methods here. So AlphaFold 3 through to Boltz 1. Yeah. Now, AlphaFold 3 is actually the best at these sort of things which are dissimilar to the training set. So that's the dark blue, also the blue with the round circle dots. Yep. And you can see that might, in those low points, is about 20%. Whereas standard docking methods will be significantly higher than that. If you go to the right-hand side, though, these co-folding methods are significantly better than standard docking methods, because here their ability to use the information they've been trained on makes them very, very powerful. So if you're working on similar complexes, you want to work with these methods. You know, they're very, very good at that. The best line on here, if you're interested in that, is actually if you took all of the results out of all of the methods, this is how good you could possibly be. So you could actually get slightly better, but you wouldn't necessarily know which one was correct within that. So I'm going to finish there and say, I've told you just 2 very short stories. There are many, many more stories I could show you about the power of being able to generate new molecules in a pocket, about the power to be able to create new ligand interactions, about the types of data that are really needed to power this. But really, I'm hoping those stories show you both sides of the coin we have here. AI can do amazing things, and in some ways, I think it can do more than people are ready to accept. You know, I wasn't quite ready to see that the large language models with no extra training could just be incredibly helpful if you just type your data into them and ask. And then you can use it as a start point, even if it's got it wrong. And the minute you allow them to have access to programs and other things, they become incredibly powerful for the processing of lots of things we have. But on the other side of this, quite frequently these methods can be just beaten by very standard traditional methods. And often we don't compare and test in ways that are fair or ways that actually demonstrate when these methods will be useful and when they will not. And so the true predictive ability is not always obvious, because they are often very powerful, the new methods, but they are powerful in particular cases in particular ways. And I think everyone wants to scream they're useful for everything. And actually they don't have to be. We just have to understand where they are useful. So finally, I should say a massive thank you to my research group, most of whom are in that photograph there, and a thank you to all the organisations across the bottom of that slide who quite literally give me money to do the job that I love. Thank you very much.
Thanks, Charlotte. That was brilliant. I took away, use the right tool for the right job and understand your toolbox. So yeah, I think that's a very key message in this field. I think we're going to have questions at the end if you can stick around. But I think for now, thanks a lot for your talk, and we'll move on to introduce our second speaker, Dr. Olivia Becquignon. Olivia is an assistant professor in AI and structure-based drug design at Leiden University. He's an assistant professor in AI and structure-based. Oh, sorry, I said that. Within the university's Department of Medicinal Chemistry, he holds a PhD in computational drug discovery and a degree in biotechnology engineering. And after 2 years postdoctoral position at Amsterdam UMC, where he studied the blood-brain barrier permeability of kinase inhibitors for glioblastoma, his research now focuses on combining AI, chemoinformatics, and molecular modeling. And among his significant contributions are the Papyrus bioactivity dataset, which has been downloaded more than 15,000 times, and several open source tools for drug design. So I will hand over to you now, Olivia, for your talk.
Well, thank you very much. And I'd like to thank the organizers for the invitation to present the research that I'm doing, and particularly Professors Koppel and Mifsud for their really kind invitation. So indeed, I'm going to talk mostly about the results that I obtained during my postdoc at the Amsterdam University Medical Center on modeling the blood-brain barrier permeability of small molecules. So yeah, I'm a researcher, so well, I have nothing to disclose. This adventure, let's say research adventure, started when I joined the neurosurgery department at the Amsterdam UMC, where they focus mostly on researching treatment for glioblastoma, which is a disease that is associated with a very grim prognosis. It is the most common type of primary malignant central nervous system tumor, meaning that it's not a metastasis, right? It's a type of tumor that is developing in the brain, and it has very low incidence with a bit more than 3 in 100,000 cases, but it affects mostly older people, where if you have a look at people aged above 75, then the incidence rate increases to 15 in 100,000 people. And as I said, it's a devastating disease, as the average survival is between 12 and 15 months with the current status, well, with the current treatment, which is what is used. And the reason for that is because we're dealing with mostly cell heterogeneity that is difficult to tackle. So different cells in the tumor itself have different degrees of aggressiveness, growth, and of course come with different levels of therapy resistance. Additionally, there's a spatial heterogeneity of the tumor itself. So in the contrast that you can see here on the T1 contrast, you can see here on the right-hand side, then the bulk of the tumor, the inside is hollow. It consists only of fluids, right? So you cannot really resect that part. And what really matters is really the border, the margins of the tumor, where indeed we have the most aggressive types of cells that expand in the brain tissue. And lastly, this is a very invasive type of tumor, where the cells actually infiltrate into the brain tissue. So doing a small resection of the margins only is not leading to, well, it's not, let's say, taking out all of the material that is making up this tumor. As you can see on the flare that is on the right-hand side also, you have indeed these invasive type of tissues there. And I mentioned the current treatment, and that is a treatment that was developed in the early 2000s. And it consists of both resection, so excision of the tumor tissue, but also of combined chemotherapy and radiotherapy, of which the chemotherapy is temozolomide. And that is a prodrug, meaning that it is administered as a drug, and it makes these ions, these methyl diazonium ion that you can see here on the right-hand side of this reaction pattern. And they create those methylations on the DNA of the patients, so on the cells of these tumors that are in the brain, and making these kind of alkylation patterns on the DNA leads to cell death. Unfortunately, only 5% of these methylations lead to cell death. And there's also an enzyme that is the methyl-guanine methyltransferase that indeed excises these adducts that are formed by this methyl diazonium ions. So we see also a mechanism of resistance, especially in patients that overexpress this MGMT protein. And of course, as you've understood, then this agent, these methyl diazonium ions, need to get into the brain to reach really the tissue of interest, right? And they have very low BBB permeability, so their penetrance in the brain is rather low. And I'll come back to these values that we see on the screen later. But I want to stress that it's not only the case for temozolomide, but for many of the drugs that we use, especially if we look at the marketed drugs, especially we see that only a fraction of them are able to get into the central nervous system. Meaning it's very difficult to address this and design molecules to get in the brain. And the major barrier there is what we call the blood-brain barrier, and I'm just going to talk a bit about that so that we're all on the same page. So this blood-brain barrier is actually this line of endothelial cells of the blood vessels in the brain, and it delineates what makes the blood from the brain tissue. right? And they prevent the entry of a lot of molecules in the brain tissue, which is really nice if we're thinking in terms of toxicants, but it's not as nice if we're thinking of drugs, especially, for instance, in terms of temozolomide, for instance. So should I consider a drug that gets into, let's say, the blood, then the entry into the brain tissue would be conditioned by the permeability of these molecules, right, through the blood-brain barrier, so through these endothelial cells. And there's a mechanism called passive diffusion, in which the molecules that are in the blood, the medicine drugs, dissolvate and pass through the membranes of those endothelial cells and resolvate. But this is conditioned by their physicochemical properties. And I'm not discussing now the active transport mechanisms that can be also at play, that either pump the molecules inside of the endothelial cells and in the brain tissue, or efflux them outside, so that you either sequestrate the molecules in the brain or just keep them in the bloodstream. But if I'm going back to trying to improve molecules for their BBB permeability, what we can have a look at is the ratio, the concentrations between what is in the brain and what is in the blood. And this is this log BB ratio that we're looking at. And of course, the higher this ratio, the more of the molecules is accessible, is in the brain, right? And where this ratio, because it's a logarithmic scale, is if it's greater than 1, then it means that you have 10-fold higher concentration in the brain tissue, while if it's lower than -1, you have 10-fold lower concentration in the brain tissue compared to in the blood. I'll come back to the fact that this is looking at only the total concentration of drug, also considering drugs that are bound to plasma proteins, as highlighted here on the slide, that are not free to move in the brain tissue, but also the drug in the brain tissue that are actually bound to brain tissues and not freely moving around. Anyways, when we are looking into how we can model the blood-brain barrier permeability and how these molecules get in the brain, we're faced with quite a few challenges and other barriers, as I call them in the title. Mainly the scarcity of quantitative data. And if we have a look then at qualitative data, and here's a little survey of the publications that focused on the predictions of what would be a blood-brain barrier penetrant and what would not be, then there are many, let's say, issues that we can find already. So first, there's a wide variability in terms of the number of molecules that these sources use their AI model to train them. Secondly, you can see that the data sets that are used are usually quite imbalanced, sometimes in favor of the BBB permeable molecules and sometimes for the impermeable molecules. And thirdly, the thresholds that are used are usually close to -1, meaning that anything that has a concentration that is slightly higher than less than 10-fold concentration in the brain rather than in plasma is considered to be blood-brain barrier permeable. This is also not considering the variability in terms of the experimental methods that these data points rely on, because they usually aggregate a lot of data that comes either from assays that are done in different strains of rodents or even across different species of animals, and not considering either the translational aspect to human. And finally, as you can see in terms of the accuracy of these methods, these classification methods, they claim to reach 98% accuracy. And this should, let's say, already tickle our brains, right? Which at least it does to me. When looking at that, I'm thinking, oh, either it's a solved problem or the accuracy that is measured is probably on a data set that is a bit too easy, relating to Professor Dean's type of evaluation, where there's maybe something is going wrong there in terms of these evaluations. So in order to address this, these two effects of data scarcity and lack of molecular diversity, well, one could say, okay, I'll go without even considering them and train a type of AI model from the experimental data that is available, the quantitative data that is available. And this is what one would usually have. So this is kind of the baseline model that I'll refer to as if I train a simple AI model, which is consisting of decision trees that kind of classify the data and in different small bins so that it's able to predict a continuous value. And here MAE stands for the mean absolute error of the model that we see. So we want to decrease this error. RMSE stands for root mean square error, which is also an error, but it's actually, as it's a square error, it addresses also what is really outliers, right, and adds more weight on those. And on the right-hand side of this table, we have the R square, which is the coefficient of determination that tells us how good is the model in predicting compared to ground truth. And you want that to get the highest and the closest to a value of 1, which is a perfect prediction in that sense. And you can really see that, well, there's quite some error, right? 0.5 log unit in terms of absolute error on average, and a coefficient of determination that is rather low for what we want. So one way that we can then consider hacking the data scarcity and lack of molecular diversity is, well, obtaining more experimental data. And that's extremely difficult, right? And it's a very onerous type of experiments. So this is really difficult to do for large data sets. So one would consider probably, let's move to a next step. And instead of obtaining true experimental data, obtain proxy data that would be good enough for our prediction systems. And this is where, let's say, the fact that I live in the Netherlands kicks in, because I don't want to reinvent the wheel of a bike, right? And I find this patent quite fun, that it's been approved in the late 19th century. So what I mean by that is that instead of relying on proxy data, what we can do is actually rely on the plethora of different AI models that have already been trained on in terms of predicting the BBB permeability of molecules, right? And the way I see them is that each of these different models has been trained on a very specific subset of what we call the chemical space, so all the possibilities of the distributions of physical chemical properties of these molecules. So if we somehow train from all of these prediction systems, maybe we would get somewhere close to the experimental data. So it would kind of get us closer to better predictions. So then, instead of training a model, the first challenge becomes in obtaining predictions and on a large set of molecules. So what would be a large set of molecules? So thankfully, we were able to gather data from the Papyrus dataset, which is a collection of publicly available databases of experimentally obtained molecules, but also from compound providers and other datasets, which gets us shy of, let's say, a bit over 6.5 million molecules. But you might consider that, well, is it enough just to train on prediction and it's reinventing the wheel, right? Then you'll get just a prediction that is trying to predict what was already predicted. So that's why we need to do this second adjustment on true experimental data. And then we need to gather a data set of experimental data. So a small survey of the literature gets us to around 33,000 molecules with duplicates. Right. And then comes the issue of processing such data, because here I must stress that all these values consider duplicates. And molecules, either by resonance forms in which double bonds can kind of switch where they are located in a molecule in conjugated systems. Tautomers are also a mechanism at play in which, and especially for protomers, where hydrogen atoms switch, let's say the atoms that they are located on, leads to quite some duplication in terms of molecules, where the structures appear quite different, but what is actually measured in vitro is rather different. So we need to kind of deduplicate all this data. And thirdly, for only the experimental data, not the prediction parts, but the experimental data, we want very high quality data, right? That the data is quite accurate. So in that sense, what we did is removed any high variance between multiple measurements. So should a molecule have been assayed multiple times and the variance of these different readouts be greater than 0.1 log units, then these molecules were disregarded. And this doesn't drop the number of molecules, the unique molecules that we get in terms of the large data set for which we'll obtain predictions, but it does a lot for what has been assayed experimentally, where we go from, let's say, shy of 33,000 molecules to a bit less than 800 molecules. And if you remember, I told you that, well, the models, the AI models that have been built, let's say, to this date, usually do not address that the evaluation of these AI models should be rather robust and test how far they can extrapolate to new molecules and not just be interrogated on molecules they have either already seen, for instance, duplicate molecules, or molecules that are extremely similar to their training data. So that's why we do a bit of a clustering in which we extract, we separate these 800 molecules into two subsets that are very distinct from one another, where the Tanimoto distance between these two sets, any molecules in these sets, is greater than 0.65, meaning that we can use either of these tests, these subsets, and really ensure that the model has not seen something similar during its. So when it comes to all of these barriers, as I call them, well, by using a large data set, then we've addressed the data scarcity issue and the lack of molecular diversity. And by the clustering method that we've used to split this subsets of experimentally determined values, then we address the last part. And then you might consider, well, is the last part, like training on experimental data, really valuable? And on top of just not predicting already predicted data? Well, you can already see that if I look at the predicted values of what I call the training set, right? So this very large sets, the predictions, the distribution of the predictions is quite far from the values that we get out of our experimentally determined values, right? And this is also the case in terms of lipophilicity of these molecules, but also in terms of molecular weight, where we see quite a, let's say, a variance associated with this large set. So to stress again, what we're doing is what is called transfer learning, where we first train, you could see it as training an LLM, right? A large language model that is trained on, for instance, many books, and then you fine-tune it on very specific tasks, for instance, elucidating NMR structures, as we've seen in the previous talk. Here we train them on predictions, so previously established models from the literatures that will predict the log BB ratio. And then we continue training them, but on only one subset of true experimentally determined data. And finally, we assess the performance on this dissimilar subset of experimental data. And you can really see the distribution there in this human plot of these molecules, which tries to plot in 2D, let's say, all the different physical chemical properties of these molecules, where you can see that the molecules from the test set are close to any other molecules, either from our training set or fine-tuning set, meaning they are relevant for the task that we want. But because, as I told you, we have established this threshold that polarizes and limits the similarity between them, then we're really sure that it's a fair assessment. And we trained different types of models. So the first type of model was the baseline model, as I told you, trained only on experimental data. And then two other types of models, one where we do this transfer learning, so we train on predictions, adjust on experimental data. And then the third type of model is where we do not use molecular features like physicochemical properties that are predefined, but that let a model come up with its own features. So I won't go too much into details there, but it's a model that is able to associate specific atomic features and bond features that for which weights are trained during the training of the molecule. And each of these atomic features is aggregated with that of the atom's neighbors, so that you get what we call the molecular embedding, which is representative of all these atomic features and how they contribute to one another and how they are neighboring one another. And so that you could train from these to predict the property here that would be the log-BB ratio. And all of these three models were trained in three different schemes. First one where we train only on the log-BB ratio, right? So the ratio of the concentration in brain over plasma. And then one where we add quantum chemistry derived features. So they are the energy of the highest molecular orbital and lowest unoccupied molecular orbital energies. So it's just adding a lot of non-linearity on the data, because these properties are, let's say, rather uncorrelated from the standard molecular descriptors. And the third scheme that we're using is adding pH-dependent solubility values at three different pH, acid, neutral, and basic pH, where these physicochemical properties are quite important for the possibility of the molecules to desolvate, as I mentioned, close to the endothelial cells and resolvate, so mainly capturing these physicochemical properties for passive diffusion of the molecules. So if I compare the different types of models, so the baseline trained only on experimental data, transfer learning simple model, and a model that comes up with its own descriptions, what we can already see training only on the log-BB data is that moving along these types of models, you see already a decrease in terms of the error associated with models, right? Where we reach around 0.44 log units of the error. And we see also the same with the coefficient of determination, with a drastic increase in terms of the coefficient of determination. So we're getting way closer to ground truth. And good to know that it's a very difficult set, right, that we talked about. If we have a look at the, let's say, at including quantum chemistry derived features, so adding non-linearity, right? So we're still, meaning that we're training the model to predict both BBB permeability, but also the energies of these molecular orbitals, and only evaluating the model on log-BBB data, we already see that there's further improvement in terms of the error, right? Where, well, the only training on experimental data is doing worse than indeed doing this transfer learning or this improved type of model. So this means really that we're really improving in this type. Though you could say that, well, these two models seem to perform similarly, right? Between the two ChemProp models, whether trained on 1 or 2 or 5 tasks. And if we further include pH-dependent solubility values, then you can really see that a drastic, continuous improvement and a decrease of the error that is associated to these models, and an increase in terms of the coefficient of determination. So if I talk in terms of Pearson correlation coefficient, these values, like of the last model, of the bottom model, which is 0.72 in terms of Pearson correlation coefficient, which is very high for a data set that is completely dissimilar from anything that the model has been trained on. So that leads me to a conclusion there. That is that by using this transfer learning, and you can think of not only in predicting log BB ratios, but any other any PK data, really, right? Then that we actually were able to mitigate data scarcity and lack of molecular diversity by using a very strict way of assessing the models. We're able to carefully design methods and really test how well they would extrapolate to new molecules, and that's really the case that we wanted to raise there. Through the different metrics that I showed you, you see that really that training from experimental data gets you, let's say, something that is not as near to the ground truth as when using this pre-training step on predictions and continuously learning on experimental data. And one key aspect to remember is that training on multiple types, so training a model to also try to capture more than just the readout that you want to predict, gives you also an advantage and a better performance. Of course, this doesn't consider what I mentioned earlier, the free molecules that are able to go on the site of the tumor, right? So it's taking into account all the molecules that are bound to plasma proteins or brain tissues. So maybe we could also use these AI models to, let's say, capture the amount of the molecules that is bound to these tissues and indeed translates to the unbound fraction of these molecules. And maybe we could use such type of models to actually help with PK/PD modeling, where, for instance, we could use predicted parameters, such as the ones, for instance, that we predicted here, to help in getting PK/PD models translatable to many more molecules, right? And of course, as I mentioned, it doesn't take into account yet plasma protein binding or brain tissue binding. It doesn't consider either the polarization or the distribution of the drug in terms of inside of the brain tissue itself, or where the tumor is located. And it doesn't consider also the distribution of the drug inside of the tumor itself, right? Where you can see different concentrations in the tumor itself. But hopefully we will get there. And with that, I'd like to acknowledge all the people that helped in getting these types of models for very difficult tasks. And with that, I'd like to conclude my talk.
Thanks, Olivia, for the great presentation. I think showing there the requirement for a lot of data to make a good model, you need the data, and how you've sort of overcome that, and how you might use these all knowledge available to you to make the best model available. So yeah, I think thank you very much for your presentation. We've got a little bit of time now for some questions. So if you've got any questions, put them in the Q&A. I think Charlotte, I think she might have left the meeting. She did have to leave for some other business, so Charlotte might have left. So Olivia, you might be on the spot here, but yeah, so if anyone's got any questions, please put them in the Q&A section. So there is one question there, which is pretty broad and quite a difficult one, I think, to answer. I mean, I don't think I would feel qualified to answer it, but it is around how much resourcing from third world countries is being put into annotating and training large language models. So how much there's a big human annotation effort to be building large language models, and how much of that is coming, I suppose, from the third world. So it seems like a pretty broad topic in terms of it's leading onto ethics and all these other sort of questions around AI. I did ask Charlotte about this just as she left for a little for a comment, and she said it was this effort is to improve the quality of the data, but the annotation's quite a tiny part of the data that these large language models are taking in, and the internet has billions of pages indexed, and the size is probably 1 trillion, and the pages and that's pages, not words, concepts, and pictures. So I think what she's saying is there's also a lot of other data coming in from sources that are already sort of annotated. So that's only one part of the picture. I don't know if you want to comment on anything.
Yeah. I can add on that. That's a lot of these large language models, as they exist now, are not just one massive model, right? They consist mainly of also some specialized sub-models that are called by prompts, right? And the AI companies, try to actually get really well annotated data to train these sub-models that are very specific in terms of what they can do. So I would expect, indeed, as Charlotte mentioned, that the NMR assignment and the model being able to decipher really the chemistry would not be carried out by the large language model itself, but by a kind of the AI annotation itself that comes from, let's say, scraping the World Wide Web, but acquiring first the really well annotated data keeps the ball rolling from the beginning, right? And as then the first types of models get trained, they can also themselves then further annotate other types of data that they can get trained on and on and on. So I would say it keeps it gets the ball rolling, at least at first.
Yeah, so it's an important aspect, and who's doing that and how they're getting remunerated is obviously important as well. But maybe this is a sort of a wider question for society, and there's obviously other parts of science where we have to think about the ethics of what we're doing and how we're achieving those things. I'm not sure if we'll be able to answer that today, but yeah, so we've got a few more questions coming in now. So I think the first one that I've got here is kind of the billion-dollar question about building machine learning models. Of what data set size do you need? So I work in the chemical industry where data is much more sparse compared to the pharma industry. So I don't know if you want to comment on that, Olivia. Obviously, you talked about some techniques as to how you might improve your data set sizes.
Yeah, so we've had experience actually. First, let's address the first question in terms of what is the size needed. The easy answer is the more the better, and it's not meaning only 10 additional molecules will do a difference. It's really in terms of orders of magnitude, right? So if we really look at it, the experimental data that I presented was just in terms of the shy of 1,000 molecules, let's say, and we're training really on millions of molecules, so orders of magnitude bigger. With internal experience here at Leiden University in terms of sparse data, where we try to use models to, let's say, complete the picture, where we first have small dedicated models that are very good in terms of filling in the gaps of the data that we have, so that you could think in terms of imputation, right? Data imputation, where we would then supply the entire matrix of data that has been filled in with those specialized models to a larger type of model that could learn from, let's say, broader patterns in the data. So that's one way that we've been dealing with that internally. But yeah, going back to the original question, what size do you need? Well, it really also depends on how sparse is the data that you have, right? So it's a really project dependent answer that you're looking for in the end.
Yeah, thanks. I think yeah, just from my point of view, the applicability domain and generality of the model as well play a big role as to if you're trying to make a large language model that'll answer any question that you put to it, you're going to need a lot of data. You're going to need a lot of data to make that work. Whereas, yeah, if you're looking at one specific chemical type, you might be able to build a model that is only applicable to that chemical type with a smaller data set. But like you said, you still need a good coverage of the space. Yeah, so we've got a few more. So this one's specifically for you, Olivia. What are the next steps from this research? Are there going to be in vivo tests to assess the true predictive accuracy for some of the molecules?
Yeah. So we're currently running those tests on a few molecules, but this is going on at this very moment. But as I mentioned, it is also one of the shortcomings of such studies, where indeed obtaining, let's say, prospective validation is rather difficult, also in terms of funding, right? So it's especially for these very fundamental type of research, I would say, where it's not really focused on a specific project, but in terms of improving predictions at large. So we're currently running those validations, in vivo validations, but first step in rodents. Let's see if we get to get some clinical data also from the University Medical Center.
Okay, thanks. There's another one come here about asking about the blood-brain, the BB crossover and the efficient log BB. Is that an indication that the drug may permeate to the tumor cell as well, or if not, are you working on understanding ways to measure the tumor cell permeability using AI?
Well, so yes and no. So it really depends per tumor. So we've seen actually with glioblastoma that there is a certain subpopulation of lymphocytes that infiltrate the brain tissue to combat the tumor. I'm not an immunologist, so I wouldn't be able to tell you exactly about these lymphocytes, but they form kind of a shell around the tumor tissue, right? Because they are really attacking the tumor itself, but then it means that the molecules to get into the margins of these tumors, so the tumorigenic cells, would have to first pass these lymphocyte cells themselves. So it constitutes a second type of barrier, right? So it all depends also on the exposure that you need to reach in terms of the molecule. But it's also extremely difficult to get such type of data. Specifically clinical data, because we've observed that phenomenon of these lymphocytes only in humans so far. Yeah, so acquiring the data is extremely difficult, right? So being able to measure the tumor cell permeability is quite a challenge in itself, and that's also something that getting proxy data, as I showed so far, it would not be that of an easy task, I would say.
Okay, thanks a lot. I think that's all the questions that we had open at the moment. If anyone's got any that they want to quickly put in there, we've got a couple more minutes until the break. Ian's just come on camera. I don't know if he wants to...
I just had a quick question that came to mind as Olivia was speaking. So you've talked about the kind of scarcity of data, and I think Peter also mentioned for transcription factors that there's not a lot of data out there. So what do you see as the kind of balance between who should be responsible for plugging these data gaps? Is it academia? Is it industry? Who's going to benefit more from that? Who should put the effort in to generate the data that's missing? Or do you see the kind of opportunities for both, a bit of synergy there? Or what's your thoughts on if there are these gaps there and we need to plug them, who should plug them, really?
Well, the idealistic view and the simple answer would be both, actually. Academia and the industry should tackle these. Of course, it comes with its own challenges, right? In terms of industry, there's quite some data that industry might want to keep private, right? Though in terms of development of AI methods, and I should say algorithms around AI, we've seen the emergence of techniques that allow models to be trained without compromising let's say the data privacy, right? So that there's no leakage of intellectual property from companies inside of the models. So the data that these models have been trained cannot be reverse engineered. They have been, of course, some research papers mentioning that actually this is not 100% foolproof and that you can get back to a few of the molecules that have been developed. And of course, then companies might not be that into sharing such type of data to train models if their intellectual property gets compromised. So yeah, I think in terms of the algorithms, there's definitely a move that is made to make the efforts between academia investigating, let's say, a small number of molecules, such as, for instance, transcription factor data, available and very specific, but also in terms for companies to share, well, not to share their data, but be able to, let's say, the drug development community benefit from their data without compromising them.
Okay, thank you.
I think that probably just leaves me to thank our speakers from this session and conclude this this part of the program. So I think that we're moving now to a refreshment break from now till 10:15, and then we're going to start again at 10:15, looking more closely at drug development.
So welcome back, everyone. We're now ready to start our second session, which focuses on the role of AI in drug development and building on the excellent earlier presentations on the role of AI in drug discovery. So just a reminder to post any questions during or after the presentations using the Q&A feature. And we'll try to address as many of those as we can during the discussion section. So our first speaker in this session is Professor Patricia Munro from Queen Mary University of London. So Patricia is Professor of Molecular Medicine and is the Centre Lead for Clinical Pharmacology and Precision Medicine. Her research interests focus on cardiovascular genetics. The group uses genomics and computational methods to understand the molecular basis of hypertension, cardiac arrhythmias and heart failure. And Patricia has established and co-leads several international complex trait consortia with these collaborative networks, leading to the discovery of thousands of genes for disease and cardiovascular risk factors. So Patricia is going to tell us about use of UK Biobank for discovery of drug efficacy biomarkers. So Patricia, thank you, and over to you.
Thank you very much for the introduction. I'll just pull up my slides. Hi, can everybody see the slides? Can you hear me okay?
We can see your pointer as well.
Oh, I have a pointer. That's amazing. Brilliant. So I'm just going to check that's fine. Okay, so thank you very much for inviting me to give a talk at this meeting. And as you've indicated, I'm going to really just be focusing on UK Biobank for discovery of drug efficacy biomarkers. So this is an overview of the presentation. Very much want to, again, not knowing so much over the audience how much you know about UK Biobank, what it is, the design of it, and key, what biological data sets and clinical outcomes would be available. And then really the meat of the presentation is going to be sort of the contributions of UK Biobank. And here it's been in genetics-driven target identification, because there's a wealth of genetic data on the whole data set. And this really has led to lots of new therapeutic or new targets for therapeutics, but also opportunities for the drug repurposing. Then I'd like to just briefly touch on big data coming into UK Biobank, the use of the contribution proteomics-driven target identification. And here it really is sort of integrating omics data sets and for understanding mechanisms of disease and target prioritization. And then at the end, just maybe some remarks and comments on the limitations and opportunities in UKB. So this audience is very much well aware that actually in the drug discovery pipeline, it really is the late stage failures that cost the most. And you can clearly see the cost of phase 3 trials is very, very expensive. So if we're able to make these much more small improvements in the failure rates of the early stages, it's probably the best place really is to improve with being able to really understand better what our targets are. So drug discovery as, oh, I'm really sorry, it seems to have a timing thing set up. Would you mind if I just stop sharing for a minute and click that off?
Yeah, that's fine. Yeah.
I'm so sorry. I've just sort of realized that it's doing this automatic jumping, which isn't very helpful. Let me just try and find out where it is. You always find these things out when not in the best possible place, isn't it? So I'm very sorry. Get rid of these timings. Okay, hopefully this will be better. Okay. Okay. So hopefully that won't jump again. So apologies. So I commented that drug discovery will always have its failures, but actually it's that timing of the attrition. And as we now know in current sort of the pipeline of drug sort of discovery pipeline, there's much more focus on being sort of increasing the number of targets that would be considered and also the data supporting their validation. And that's much being used much more to be able to select aggressively to try and reduce the chances of failure in a clinical trial. So genetics over the last 15 years or so has basically seen a huge contribution, and there's a relationship between having genetic supports for a target and also the probability of drug discovery success. And there's lots of different papers which are out there and reviews every year or so now showing that having a genetic association of the target and the associated indication indicates that there will be an increased probability of success of a drug reaching approval stage. And clearly big pharma and also biotech have able to sort of demonstrate this in a number of sort of reports from AstraZeneca, GSK and AbbVie, that basically if there is some genetic support for the target, there seems to be a higher chance of its success in developing into a therapeutic. So moving to UK Biobank, so it is a national and international health resource. It's a longitudinal study, and it's recruited 500,000 people across the UK in a period from 2006 from 2010, and aged between 40 and 69 years of age. People who signed up to be part of UK Biobank, they gave their consent to have lots of different measurements taken at baseline and subsequent visits as well for different studies. But also importantly, they provided blood, urine and saliva samples for future analysis, lots of detailed information as well, and also to have their health followed. So over time, since the study has been running a long time now, there have been sort of approaches back via questionnaires to the participant to provide more information or come back for further studies. And one of the biggest studies which UK Biobank has launched in recent years is the imaging study. And this is where they asked 100,000 people to come back. So they would have MRIs on their brain, I think it's their liver, their stomach, and their heart at that particular time. And they've launched 100,000 people have now been recruited and had images done. And actually, of the 60,000 are going to come back and have repeat imaging. So I'm hoping that's giving a bit of an overview of the data that's being collected on this amazing resource. And it really, its big aim is to build this powerful resource to help scientists discover why some people develop diseases and others do not. So I mentioned genetics is one of the first data sets that was generated in the individuals from UK Biobank. They worked with with a company, Affymetrix, to create a bespoke genotyping chip, which is the Axiom array. And this was genotyped in blood samples from everybody in UK Biobank. And subsequent to this, what we can do is, even if you haven't on your chip identified a genetic variant, using imputation from sequencing data, you can actually get the identity of millions more SNPs. And therefore, we have these additional imputation panels. And clearly the goal is to have full genetic coverage. In an industry consortium, they also then came together and recognized the value of not only having genotype data, so that isn't all genetic variants being identified, but also sequencing the coding part of the genome. So this was completed in 2022, all 500,000 individuals as well. And that really is a very relevant data set for informing discovery and development of new medicines. And then last year, again, with huge investment, government, industry, and charity, we've now got whole genome sequence of everybody in UK Biobank. And clearly that's important for understanding of the regulatory genome. So I'm going to touch across a couple of other data sets which are available at baseline. We do also have, I'm not sure why some of my slides have been truncated, but apologies again on that. But we do have a whole series of biomarkers, cardiometabolic, bone, joint, etc. And these have all been measured in everybody, and also 20,000 people at follow-up as well. And also again, just released, they've also working with Nightingale Health using NMR, have been able to measure up to just over 200 biomarkers, which are metabolites, again, here from plasma, and again, have repeat measures. So one of the final data sets which I want to mention as part of this presentation is additional protein biomarkers from plasma. And again, there's huge interest in, again, generating more and more omics data on this amazing 500,000 people. So a pharma proteomics project was set up. And again, that was a consortium of 10 biopharmaceutical companies. Here, they've used a platform called Olink, and they were able to measure the 300 or 3,000 plasma proteins. And this was done in 53,000 UK Biobank participants. And clearly, having a proteomics data set, you're able then to start making links between genetic factors and levels of proteins and relationships to disease. What was announced as well recently is actually, as again working with Olink, they now are able to measure more plasma proteins, and that number is going to go up to 4,500. And actually, amazingly, they're going to again get this data in everybody, and 100,000 people are going to have repeat samples. So let's take a little look at what are the proteins which are being measured, and actually which people in this pilot have been released to researchers. So as I said, there's 500,000 people in the UK Biobank, and for this study, they selected about 54,000 people. In this panel here, they're able to show you what are the number of analytes for each protein panel, and they're grouped according to cardiometabolic, inflammation, neurology, and oncology proteins which are measured. They also then, of course, had to make decisions on who they would measure this first set, or who would be that first data set. So they selected about 46,000 people, randomly selected, and then the consortium then selected just over 6,000 people specifically, and then they also took individuals which were part of the COVID-19 imaging study. So this paper here, which was published about a year ago, clearly you want to sort of then look at the age distribution of the people which we have protein biomarkers, and essentially they seem to map pretty well with what UK Biobank, the age, so they are older, and it's just the COVID people who had COVID were the younger individuals. But also, they also were able to show as part of this paper here that actually the people who were selected from the pharma consortium seemed to be enriched for particular diseases. So these were probably of interest to the companies. So it's a prospective study, and therefore numbers of cases are going to increase over time. So this was just a snapshot going back a few years ago. As I said, it's a prospective study. You will get increasing numbers of individuals and outcomes of common diseases, and hence you're going to have more and more power for new discoveries and analysis. So moving towards genetics-driven target discovery. So how is UK Biobank? Just to also comment, UK Biobank is you're able to apply to use the data, and maybe that might come up in discussion. And this is for researchers and everybody worldwide who have very valid research questions. So I thought I'll give some examples of some of the contributions of UK Biobank in this new target discovery pathway. So I work on hypertension as one of the areas I work in. It affects over 1 billion people worldwide, and we're totally interested in understanding what is the underlying genetic basis. So this is a timeline of sort of discoveries for genes which are associated with hypertension and blood pressure traits. And I put this timeline here just to say the launch of UK Biobank in 2012, and with then 500,000 people with genetic data, absolutely just sort of transformed the size of experiments that we could do to find genetic loci for hypertension and blood pressure. So the landmark study was back in 2018, and actually with the addition of UK Biobank, we were able to identify 535 new genetic loci. So as you can imagine, it's a substantial increase in our understanding of blood pressure and hypertension genetics. Using these big data, we now know there's huge complexity. I think we knew that there are many different mechanisms controlling blood pressure, but the genetic analysis is indicating hundreds of enriched pathways. Gives us information overlap with other traits, but also for this audience, it also is providing, depending on where we're finding the genetic variants, support for existing therapeutic targets. There's also identification, of course, of new therapeutic targets, and also potential repurposing opportunities. So as part of that 2018 paper, where we used UK Biobank, we did find genetic variants on some existing antihypertensive medications. So this is a sort of an overview of when you're looking at the genetic architecture of any trait. Clearly, you might have rare alleles, and they're normally associated with Mendelian disorders with a big effect size, or you have lots of common variants with small effect sizes. So if we look at blood pressure genetics in 2025, we've now got over 2,000 common blood pressure genetic variants, and these are mostly non-coding. We've got a number of variants which have an intermediate effect on your blood pressure levels, but we also have rare frequency variants which have bigger effects on blood pressure. So one of the next stages of what are the tools that are out there to help us prioritize what might be the candidate genes, looking at GWAS loci. So there are lots of different data sets out there and pipelines which you can use to move from a genome-wide association signal, which is essentially a very highly statistical set of variants associated with your outcome, to then understanding what is the mechanism of what is the causal genetic variant and what is the gene. So this work here, we were able to take the results of a GWAS study, which included UK Biobank, and understand there were about 1,900 distinct signals. So using fine mapping and computational approaches, we wanted to look at the annotation or the functionality of the different variants, and then by trying to assign different functionality to a variant, what gene would that be affecting? Is it in a gene, or would it be in a regulatory sequence affecting an expression of the gene? By doing this across different blood pressure traits, we were able to get a whole list of potential affected genes, And then beyond this, you can start looking to see what other data might be out there in animal models of disease, the gene and expression level, protein expression levels as well. So you can whittle it down with some confidence of how many genes which you might think to be associated with blood pressure. And as part of this work, we have nearly 400 genes which seem to be non-overlapping across the traits. And these then would be sort of a listing of ones that you might want to interrogate further to understand mechanisms, but also essentially if you wanted to see if they are in pathways that perhaps you might want to target for blood pressure lowering medications, but also maybe medications which might reduce cardiovascular diseases. With a GWAS signal, however, also to comment, even using a pipeline like this, that you don't necessarily always get what you believe to be a really good effector gene from the data sets available for you to interrogate. You can perform druggability analysis, as you can actually see if any of those proteins already have, could they be druggable to a small molecule, but also are there existing medications to some of the genes you recognized? And as part of this analysis, the answer was yes. So GWAS loci and common variants in non-coding space is actually quite challenging to work through what might be the genes and the mechanisms. However, to comment, we went through our own analysis platform, and as researchers in academia, we tend to, there's always new tools and data sets and algorithms being developed, which we will apply and create our own pipelines. But I think what we also tend to use is the Open Targets platform, which was set up by academics in industry many years ago now, and it's continued to go from strength to strength. But it's essentially a database which collates the data. But if you have a gene of interest, you really can sort of go in and use this platform to interrogate associations with disease, gene expression levels, if there are knockout models, if there are already drugs in development, and even potentially safety profile. There's also going to comment, there's Genomics PLC, which is a British tech company. They launched in October last year a multi-omics platform to help power drug target discovery and validation. So as part of their platform, again, you can bring your data into the secure environment. They've got proprietary algorithms and even data, and all that's apparent on the website at the moment is the number of GWAS studies they have and how much transcriptomic data. But this is a new sort of opportunity, again, for companies to work to understand better maybe what would be a good target to take forward. In the blood pressure world, so I mentioned we've also found rare genetic variants as well. And with these in 2016, we found rare genetic variants in NPR1. And this natriuretic peptide receptor, basically atrial natriuretic peptide and brain natriuretic peptide bind to this receptor, which lead to an increase in cyclic GMP. And we know it has effects on the vasculature and cardiac effects, and it leads to decreases in blood pressure. Work by Regeneron, they'd also contributed to the consortium using UK Biobank data, were able to subsequently validate the association of missense variants, which was shown functionally to be associated with loss of function or gain of function. So both Regeneron now and also Novartis have drug development programs with molecules, monoclonal antibodies in clinical trials for different indications. And for the Regeneron molecule, this is just data showing time after dosing, it seems to work really nicely. It also, in a canine model, reduced central venous pressure. And actually, for them at the moment, they're developing the NPR1 agonist as a treatment for heart failure. Novartis have released data and published last year in Circulation showing it's got a good safety profile that seems to be long-acting, and it's being developed for the treatment of hypertension. So I think it's sort of just showing just exemplars by having big data and genetics, how it can really inform potential new targets and also down the line using omics data, potentially new mechanisms as well. Other exome contributions to new therapeutic programs is, again, an analysis here interested in identifying genes which might be associated with body mass index. These very rare truncating variants was found in this receptor, GPR75, and essentially they were able to show that even if you had only one copy of this genetic variation, you've got a much lower BMI and a 54% decrease rate in obesity. Looking at that gene, when you knock it out in mice, again, it's got resistance to weight gain. So GPR75 inhibitors are also now in development. AstraZeneca and Regeneron are in a partnership. This G protein receptor, however, there are potential more complexities in its drug development program. Just understanding what its ligands are. I think 2 have been reported, CCL5 and 20 heat, but also genetic variation as well in this gene has been associated in many other outcomes as well. So it's associated with Alzheimer's disease, but also cardiovascular traits. I now want to move on to proteomics-driven target discovery. So this paper, which was published again last year in Cell. So I mentioned at the beginning, there is Olink data, which measures about 3,000 proteins in about 54,000 people in UK Biobank. So clearly when you have this data, you can now work through and look to see associations of plasma proteins with diseases. And this is what is published in this paper here. So you've got your about 3,000 proteins, and they've done this huge study testing of differences of protein profiles, 986 different traits, and across many different prevalent and incident diseases. Of course, they've demonstrated there were differences across different traits and diseases, and then you can do downstream analysis to understand what might be the biological pathways. What they also were able to demonstrate, if they're picking out proteins which are differentially expressed between cases and controls again, can they then use these proteins to predict disease? But also the other area is looking to see if you have and work through, can you sort of work through whether that protein which is thought to be associated is a potential drug target as well. So this paper is really interesting, full of information and data, and actually also were able to illustrate lots of pleiotropy, so many different proteins involved in many different disease traits and mechanisms. So that was really interesting. So as an exemplar, I just wanted to say, just to give an example of one paper, and this was work by Donahue et al., and this is published again in Cell Genomics last year, and this is from Genentech. They were interested in using the plasma proteomic data set to see if they can find any new insights into mechanisms causing asthma. I'm very briefly going to just overview what they did. Clearly in UK Biobank, you've only got a small proportion of people who have the plasma proteome measured, and it's on a limited number of proteins. So they defined case controls for asthma in this smaller data set, which has the proteomics. So you've got 4,000 cases and 23,000 controls. And then they wanted to have an independent data set, again, as a case control grouping. The first thing they did was to compare protein levels between individuals with asthma and those that don't, and see how many were differentially expressed between, or differential, what were the differentially abundant proteins. They found about 609, and then clearly when they've done other analysis and adjusted for covariates, the number reduced down to 269. So at this point, you're looking and seeing there are proteins associated with asthma. However, you don't know whether that protein is a consequence of the asthma or whether that protein is in that causal pathway to asthma, and thus clearly be important to understand disease mechanism, but also it might be a potential therapeutic target. So with your proteomics data and having genetics data, you actually can use genetics or pQTL, so protein QTL loci. You can know and understand what genetic variants are associated with levels of protein expression. And actually, if you have that type of data, you can then start working to look and see, can you test genetically predicted levels of the protein and if they have any causal relationships with asthma. And that's essentially what they're able to do. So they sort of went back to the whole proteomics data set and they worked out genetic variants that would be mapping and associated with levels of the expression of those proteins. And then they worked through those essentially to find out that they did find genetically predicted protein levels associated and gave us some causal mechanisms. And other parts of that work, they were able to show some of the proteins that they found are already targets for drugs which are in development for asthma, but they also found some new relationships as well. So to end, there are limitations to UK Biobank. It's predominantly white British, therefore it limits transferability of findings and maybe translation medicine opportunities. There's no recall for consenting, so you can't go back and bring people in for additional studies and clinical studies, for example. Rare disease is underrepresented, and that's just by nature of the design of the study. They're older, healthy, and wealthy. As I mentioned at the beginning, there's quite limited tissue sample, and there's a reliance on electronic health data, but actually there is some linkage to GP records, and hopefully that's going to extend. However, across the world, UK Biobank is just one biobank. There are lots. And basically, a lot of these is just, again, from a review, it gives a bit of a snapshot across the world, different biobanks now out there. And two of them I can just comment on, which is the All of Us study in the US and our Future Health in the UK do actually have permission of consenting, but also there's a much bigger increase in ensuring diversity has been part of the recruitment process. Opportunities, clearly it's longitudinal. There's always release of new data, the health code records. We've got imaging data. There's much bigger proteomics data, etc. And I didn't cover here, it also is a resource for pharmacogenomics and doing other studies and work too. And I just want to just flash up a slide. I work in lots of UK biobank projects. And clearly, I just wanted to, as I said, just to highlight who the team is. Thank you for listening. Sorry, I ran over with having to go back again.
No worries. Thank you, Patricia. That was a really interesting overview of UK Biobank. And I already thought of a few questions about how that might kind of interact with the developments in AI, ML, which maybe we could cover in the discussion section later if you're able to stick around after the next presentation. But,
yes, I will. I think you've shown. Brilliant. Thank you.
Great. So yeah, if anyone has questions that have sprung to mind as Patricia has presented there, please put them in the Q&A. But for now, we'll move on to the second speaker in this session, and then we'll come back together for a joint discussion section at the end of that. So our next speaker is Dr. Srijit Seal, who is a visiting scientist at the Broad Institute of MIT and Harvard. Srijit obtained his PhD at the University of Cambridge here in the UK and completed his postdoctoral training at the Broad Institute of MIT and Harvard in the US. So Srijit specializes in machine learning and cheminformatics. His research focuses on developing machine learning algorithms for drug discovery, particularly in toxicity prediction. He's a fellow of the Cambridge Philosophical Society, and he serves on the board of directors of the American Society for Cellular and Computational Toxicology. So Srijit is going to talk to us about making machine learning successful in drug discovery. Srijit, if you're there, over to you. Thank you for your participation, and we're looking forward to your presentation.
Yeah, so you can hear me, right?
Yeah.
Okay. Yeah, thank you so much for inviting me here. Yeah, so I did my PhD in chemistry and machine learning particularly, and I wanted to talk about how do we really implement machine learning in drug discovery. So let me go in. Yeah. So I'm a computational chemist. I'm not really a drug discovery expert in any way. I'm mostly machine learning from my end of side, but I've been working with drug discovery for a few years. And we had the statement that said data is the greatest drug of the 21st century. And machine learning enables this data driven drug discovery to a large extent. So a few months ago, we actually got together with around 15 different pharma companies and a few academic institutes and even some startups to write a paper about how machine learning can be used for toxicity prediction, which is on target, off target effects, but also just to look at in vitro, in vivo assays and experiments, respectively, and to see what really drives success in the real world. And how do we implement this? So this paper is accessible. You can take a look at it as well. And let's start about how this all began. It began in the 1981. Fortune magazine announced the next industrial revolution of designing drugs. 20 years down the line, there was a lot of interest. It slowly died down, but again, now with so much computational power being available in the last few years, it's really picked up again. And one thing that really matters is what industry realized is not to try and save the time or the quality or the cost of drug discovery, so not to get cheaper ingredients or not to speed up and make people work longer hours. That doesn't really drive the net savings compared to a baseline. What drives saving is if you have a good drug in the first place. If you can reduce your failure rate by 20%, you really make a lot more in savings in the other pillars. And this is how AI in drug discovery really picked up. The the basic idea is a compound has a primary activity on target. It also has a various physical chemical properties, PK/PD, metabolism, chemical reactivity, secondary targets, off targets, etc. And overall causes a physiological effect. Drug discovery, however, works the other way. So you find the target and you screen on this target. You consider the other activities and other properties in a multi-parameter optimization problem, and together you try to address an indication with the physiological effect it causes. And because of this reverse flow of drug discovery, the reverse direction, we have to be careful about how we implement any method. And so Andreas Bender and Isidro Cortes Siriano at EMBL and University of Cambridge came up with this pyramid of drug discovery, where we look at the question and hypothesis first, then at the data that's available, then the representation to capture relevant information, and finally the method that will do this capturing. A method of a very fancy method you see on the internet cannot save an unsuitable representation if you're not really representing your chemical space properly. That cannot remedy relevant data if data is wrong in the first place for an ill thought through question if your hypothesis is wrong as well. So it's not all the fancy models that really matter at the end of the day. The problem in generating the hypothesis is to look at the identification of key parameters, readouts needed to answer the question and that are practically relevant. So in vitro, in vivo assays, they must have relevance to human toxicity. There's quite a lot of push now to replace animal experiments. And so the reason is to have more relevance to human toxicity primarily, to have mechanistic insights, and the assay must be large enough to cover chemical space and translational potential. The proxy endpoint, therefore, that we have, for example, the hERG inhibition assay, must have strong correlation to human toxicity readouts. And in this case, sometimes you don't have evidence of absence, you just have missing data, so you have absence of evidence. And machine learning models need to treat these differently. It's basically to say not to replace empty data with negative data. So the idea is to have a semi-causal models, not just look at the endpoint of interest, but also look at the various other causes or mechanistic reasonings for behind that assay. And we did one of these with Dilly model and this one with cardiotoxicity, for example. Instead of just predicting binarization or dichotomized values, we try to look at at continuous regression values for the various mechanisms that cause cardiotoxicity. And so Drug discovery is not only multiple optimization across the properties, also to minimize the risks that I showed. And these risks need to minimize, and the machine learning models need to be made more reliable. And to do so, we want to figure out 5 pillars that make machine learning models more reliable in this work. And we start off with data in the first place, and we need to find if data is relevant. Choosing the right data sets is generally priority. We try to look at relevance to human biology, which means we have binding assays, cell-based assays, microphysiological systems, all the way to in vivo studies. It's easier to understand as we go up the ladder, and it's more difficult to predict as we go down the ladder. Binding assays are generally one of the easiest ones to predict among these 4. We need to answer relevant questions because a drug could have altered cell organelle morphology, gene expression, protein levels, protein activity, metabolism, and we have measured readouts understanding for each of them. So we have omics data sets like imaging data, cell painting data, transcriptomics, proteomics, metabolomics, and we try to inculcate this data into our models directly along with chemical structures to inform decision making. And that's the other representation part of chemistry, and I'll go into it in a bit. Suitable data and amount of data, we need more dose dependent data. Generally most data sets out there in the public are binarized data with 0 and 1. We need more dose dependent data in public domain to make right data sets. And suitable biology, so that's what I was saying as well. We don't really have a lot of details on IC50s or continuous regression variables in the public domains. There's a lot of simplification, benchmarks, fancy models being published on standard benchmarks that are not well curated. So let's look at the next part. So that's the representation part. So we want to see how do we ensure chemistry is properly represented in ML features. They need to have diverse molecular information. Not all molecules are the same. Representing a cat's image is very easy. It's largely unconditional. It will always be a cat no matter which angle you look at, how the brightness changes, etc. A molecule, on the other hand, is not the same. It's a very conditional label dependent on the assay, the endpoint, the dose, and therefore it's not easy to represent them. You can represent them with physicochemical properties. We can use a large number of models, and largely this approach is trial and error and not intrinsic to chemical domain. In the biological domain with omics datasets, it's even harder. And this is the representation can range from proteomics, transcriptomics, high content imaging, epigenetics, histopathology, etc., etc. Again, heavily conditional labels, and in particular, the information content of these biological readouts has only been established in particular cases. So we need to figure out still which one works for which endpoint. We ensure chemical fingerprints, for example, molecular information in fingerprints work well for small molecules. But when the molecules become larger, you literally have every fragment in each of them almost. So we then look at 3D descriptors, which work really better for natural products. So we really need to think about which features we use when, and it is challenging to select these features and decide how to train the machine learning model. We need descriptors, therefore, that mirror real world images. So we need chemical, biological data. Chemical data generally are fingerprints, properties, 3D, 2D, etc. Biological data can range all the way of omics datasets from gene expression, cell painting. You can even have Cmax data where available, mechanisms, gene ontology, etc. And they need to mirror real world challenges. They need to we need to pre-process a wide range of these modalities and train the model in such a way that we don't leak information, but we improve the applicability domain of these models. And so one way to do that is to learn representations. You learn through the atoms as you go. I'm not going to go into too much details about this, but there are models like ChemProp that can do this. But another way would be to go and look at the biological space. So look at the chemical space through a biological lens. And here, example, we take healthy cells or disease cells, patient cells, and then we perturb them with different dyes and we image them, and you get the area, shape, size, etc., across a range of different organelles. And then that features can be used to cluster more compounds and downstream analysis thereafter. So the idea is then, after you've covered the data set and a representation, how do we build a better model? And selecting the best algorithm to learn data patterns is generally a challenge because of the data complexities. So some models are universal approximators, some fit to a particular function. And if you overdo or underdo things, you end up with a overfit model or a low expressivity model. The better idea would be to stay in high expressivity or parameterized models. And the idea here is, as we move down the curve, the bias is reducing as the complexity increases. And therefore, we need to find where to stop. If we do end up continuing, we will end up overfitting the model and increasing the bias. So we do have a nice spot to stop. In large language models where data is in tremendously large amounts, we have seen double descent in that, meaning if you keep increasing your model complexity, at some point it still starts end up going down again. And that is where large language models and large models come into picture. This observation is rarely seen in drug discovery, so we generally stop somewhere here. Um, and of course, the computational costs keep coming down every week these days. So that's becoming less of a parameter since the time we started working on this. So let's look at, yeah. So the idea of machine learning model, therefore, is to take all these descriptors, learn some relation between them to predict the property. Descriptor being the chemical structure, the property being the in vitro, in vivo effect. And to validate these models to so we need to look at reliable predictions. Am I leaking the model? Am I leaking information? Minimize overfitting and bias, like I mentioned before. Have prospective validation that needs a real-time validation, which means you don't predict yesterday's weather, you predict tomorrow's weather always. And discovery datasets are always prior. So we'll go into that. And experimental error versus noise. So we look into a different ways of trying to optimize drug discovery. 4 different cases here in the paper as well. It's written in much more details that we need to stop at various points on this curve to optimize our models, depending on what we really want to use the models for. And each point is different for a different goal in drug discovery. So, like I said, retrospective validation, prospective validation is also pretty critical, because most drug discovery doesn't work if you just try to predict a drug that you already know. So to build a model today and keep evaluating for the next 1 year and see how well it works on new chemical space as medicinal chemists design more molecules, that's prospective validation, temporal validation, that we also tackle a bit in our paper on understanding biology in the age of artificial intelligence. And the last pillar here is to have actionable follow-up decisions. So we just make a prediction that's human relevant. How do we take that forward? And one way to do that is to have more and more mechanistic assays to support your hypothesis using NAMs. So these models then increase the applicability domain of the molecule, and we don't just have scores, we also have confidence of the model. We know how confident the model is, and then machine learning experts and toxicologists can work together to make decisions. For example, a drug for a chronic disease has a high risk for cardiotoxicity, and therefore the next assay to run is an in vitro cardiomyocyte assay. And that's decision making that's happening with machine learning models. It really depends on user adoption, and a lot of my time is spent talking to experimentalists on how to use these models best to drive decision making forward. And therefore, I'll end with this. You have an input, you have a model, you have a prediction. Most machine learning models you see will talk about how great the predictions are, but there's always a context which is ignored because there's a benchmark data set that nobody adds the context to usually. But there's a disease, a target organ, a dose, a Cmax, and there's a context of the program where the assay came from. There's also follow-up assays and decision making that happens, and that's basically, I have predicted, I have a good model, AUC, balanced accuracy, whatever F1 score. But what do I do now? And that's a decision making that comes into it. Improving a model in a paper, a publication, is probably just the 3 input model prediction. Improving drug discovery takes the entire context of programs. A lot of these agentic workflows now try to address both of these sides. I'd like to thank all the people who have helped me throughout my career in designing various of the work that outlaid here. We have a chemical space that's training data. We have validation space. We have external space, but we also have to realize that the next project is in a completely different space, and that's where machine learning really brings up. So let's not fall into the trap of building machine learning models just for the sake of it, and let's step back and think about how to improve drug discovery with machine learning, and that's the goal of my work. Thank you so much.
All right, great. Thank you, Srijit, for that very interesting work to hear about. So, yeah, if anyone has questions for Srijit or for Patricia, please put them into the Q&A and we can kind of have a joint discussion session now. But I did have a few questions that sprang to mind, so maybe I can kick those off. Maybe if I could come back to you first, Patricia. So forgive me if I'm wrong, but it didn't strike me that you were kind of in the AI ML space specifically. You were kind of using the UK Biobank data to identify new biomarkers, but maybe not from an AI ML perspective. Do you get a feel for how AI ML models are being used now in kind of concert with UK Biobank data or other biobanks, and how they're kind of enabling analyses that just wouldn't have been possible before? You're muted, Patricia.
Yeah, so I think I can maybe comment just a little bit. So I am working a small bit in the AI ML space, but what we are doing, and this is what we are using in the UK Biobank, is much more on disease prevention, prediction and prevention. So a lot of the work we are doing is using data from the electrocardiogram, would be an example, and actually seeing other features from the electrocardiogram and clinical data, which we could use and as a sort of, create features using both supervised and unsupervised and deep learning methods, and then using UK Biobank as a sort of a training validation, and then taking a subset of it as a test set, and then also seeing how well our models might perform in external data sets. So we've been doing a little bit of that using ECG data set for predicting left ventricular hypertrophy, would be an example. So we are leveraging sort of the wealth of data in the UK Biobank and sort of developing different research questions in that space. Other work we're doing, again, is sort of using that clinical data, genetics data, where we might, in that instance, have created polygenic risk scores, for example. And again, we sort of plug in as much information and data as possible, and we'd be asking very sort of questions relating to clinical questions about, for example, why individuals with atrial fibrillation might develop cardiomyopathies or vice versa. And hence, again, we take our population features and try and see if we can do predictions. So that's more the space that I've been working in, and clearly UK Biobank and other biobanks permit that sort of training and testing scenarios. But in a sort of a drug discovery area, it's not something I've been doing.
I mean, it struck me when you gave the example of the kind of genes associated with blood pressure. I think you had almost 400 genes then that came out of that analysis. So it sounded as though then maybe previously investigators would have had to go and look at the functional associations with those genes, maybe on a gene by gene basis. But are any of these models now being used to maybe refine those lists of genes before you get to that kind of investigator-led?
Well, I think that is where, these big data sort of resources do enable us. There's continuously new information and data that we're using, omics data from different tissues, clearly there's metabolite data. So a lot of that, every time we run an analysis or develop a pipeline, there's a new algorithm which has been developed on data, which we know predictions. But some of the direct methods that we would use. Clearly, when we want to find genetic variation, especially now with whole genome data, we would sort of go back and start using some of the prediction algorithms of, clearly studying genes and functionality of variants and what their predictions might be. So we are using those big data sets that others have developed for us in our work, but it's not something we do. But clearly, yeah, absolutely, AI is being used for all the sort of predictions of then how we map it back to what we find as a disease-related variant, to then try and interpret it. We use what others have developed as the models of what the predictions might be.
Yeah. Okay, and then you mentioned that UK Biobank is one of many of these types of biobanks globally. So has there been any attempt to kind of combine data from those bigger biobank sets or integrate those somehow? And do you think there could be a role for AI ML there, maybe?
I think that's an excellent question. And of course, as data sets have got bigger in the big biobanks across the world, everybody's working in a trusted research environment in a TRE. So every day, is all secure data in that environment. What people are able to do, again, working across maybe specific research questions, if you've got access applications in more than one biobank, you can run your analysis. So you can do sort of more summary data work to sort of enlarge maybe sample sizes for doing things, but you're doing an analysis on one platform, but you can bring in a summary data set with limited anonymized data into the other. So, but there's no merging of, which makes sense clearly with individuals with their consent into these biobanks, what they consented to.
Yeah.
So yeah, summary statistics is what we tend to get over that model.
Okay, all right. Maybe we could come back to talk about kind of ethical issues a bit later, but I don't see any questions yet. So maybe Srijit, if you're still there, what's your thought on, as the kind of AI ML models become better and better, do you think experimental validation of what they're predicting will become less important or necessary, or do you think there'll always be a need to validate these predictions to gain real confidence in them as a kind of starting point for a next step in a drug discovery project, for example? Or do you think we could ever get to the point where they're completely reliable and we don't need any experimental validation? What do you think?
Oh, the problem we here, I think, is the chemical space is so large that you'll probably never end up with generalized models because you don't have enough data in the chemical space to train these models. So unlike speech or image generation, where there's plenty of data out there, in drug discovery, we don't have that much chemical data. What we can replace in future is more and more of the animal experimentation. Uh, but that's because these don't have a great correlation with human readouts. So clinical trials will probably be there forever. It's just that the animal experiments that we used to run as a check on these are slowly going to go down in number and being replaced with uh, more complex models, all the way from spheroids to organ on a chip models, the non-animal newer approaches that can then serve as training data to validate some of the models we train on cheaper in vitro data that we now are going to translate to in vivo endpoints. So on the chemical space side, we'll probably never end up with generalized models of chemical structures, just because we have 10 to the power 60 possible molecules almost, and not enough atoms in the universe to make all of them. But we do have an understanding that animal experiments will slowly start going down in number, and they are already going down in number and being replaced with all these NAM models that are coming up.
Yeah, okay. But from your initiative that you talked about, where you've kind of brought together lots of industry startups, did you get a feel from them that there was now sufficient confidence in many of these NAMs that they could provide the data for the training or the predictions, or is there still a kind of heavier reliance on the in vivo side? What was your feeling from that?
So I think what we saw is most, if not all, companies have invested in both the machine learning side of things and have predictive models for investigative toxicology. And user adoption in chemistry teams are ever increasing. There's feedback coming into the machine learning team to develop models that suit the workflows of medicinal chemists and toxicologists to interact with them. We do have an understanding that these models are going to go up, and more pharma companies are going to uh, have deals with AI startups. I've seen a few in the last 3 or 4 days uh, with Nabla Bio and Chai Discovery and NVIDIA taking on a partnership with Eli Lilly to start an innovation center. So more and more machine learning scientists are being picked up either as employees or in deals or in acquisitions by big pharma companies. So the higher ups also do realize that AI can really speed up the quality of drug discovery and save a lot of costs in futures. It's an investment for the future.
Okay, great. And did you get a feel from the work you did that still there is a great kind of interest or use of AI ML in more the kind of efficacy pharmacology aspects of drug discovery, or the safety toxicity aspects, or do you see kind of equal interest there across the drug discovery process?
So in my experience of working in big pharma industry directly, as well as talking to peers who are involved in such efforts, so mainly there are 2 or 3 teams which are working either together or in separate teams for on target prediction. So that's the binding affinity, etc. Then the PKPD pharmacokinetic pharmacodynamics teams, that's including development, metabolism, etc. And then on the safety of target toxicology side of things. And these three are really a joint program together. Many companies would have them separately interacting with each other, and some would have them also together to work on developing these algorithms that really optimize the parameters with each other. So there are efforts in all three domains in development side of things, and then there's efforts of machine learning all the way throughout the chain, including up to post-market pharmacovigilance.
Okay. All right, thank you. There's a question just come in for you, Srijit. Could you elaborate how to choose the right data set? How do you define right or correct in this kind of context?
So there are some blogs out there as well. I'm not going to name any particular data set, but there were when the industry was just starting up and there were tons and tons of machine learning models coming up, there were certain benchmark data sets that were released in all good intent, and these benchmark data sets were used by machine learning models to develop interest and really popularize drug discovery as a task in machine learning conferences where they have really no interest in drug discovery initially, right? They're more based on technical. They were making speech models and large language models and graph models, etc. So to popularize drug discovery, there were lots of benchmark data created at that time by computer scientists by downloading assays, and we found terrible mistakes in some of them. For example, there was one assay which we started modeling, and then somebody said, "This doesn't look like the right assay." I said, "What do you mean it's not the right assay? We have like uh, thousands of papers supporting this assay and like predicting it with 90% accuracy." And like, Srijit, it's a background noise. We detected the background noise and we put it in one of the columns. And somebody has seemed to have taken that background noise, converted into binary ones and zeros, and our machine learning models have predicted the background noise of that assay with 90% accuracy. And that's there in around 20,000 papers right now. I think every single machine learning conference paper has used those benchmarks, and one of the assays was the background noise assay. So we need to be careful about which models and which assays and which endpoints we model. At least go and look at the description and a definition at what dose. So when I predict liver toxicity, what does it mean? Is it like the adverse labels that were reported by patients? Is it the side effect data set? Is it actually in vitro assay experiments? Is it a mixture of everything? Are those two separate assays in two different conditions that have been merged together? And because most of these assays would choose the test set from within that distribution, then the test set is also not really the gold standard. So it really depends on, and that's what I meant by right data set, is to at least understand and label down what is the data set, where is the data coming from, what does it mean, before I label that I have a model for XYZ.
Okay, thank you. I guess that example shows quite nicely how you still need the kind of communication, integration of the biologist or the in vitro scientist, in vivo scientist, and the computational kind of modelers as well, so that there's that exchange of knowledge about the realism of the data and the relevance of the data. So yeah, okay. All right, there is a question that's been posted, but before we come to that, maybe I could come back to Patricia just to talk a little bit about ethics. So I guess there are kind of ethical considerations of the use of AI, ML models generally, but was that considered when UK Biobank was first formulated? Was the use of data in these types of applications and models envisaged back then? And how is it kind of considered now? I guess there's anonymization of the data, but is the kind of approval for use of this kind of public data in these private kind of AI ML models from your awareness? Or?
Yeah, so clearly I'm a user of UK Biobank, and clearly I attend the annual conferences and get the updates. So I think right at the beginning, there probably people knew, this was coming, that clearly we were going to be using machine learning models going forward. I think how people are sort of using the machine learning tools in the biobanks, the way the UK Biobank, again, just being focused on that one biobank, I think there's, as part of any access to UK Biobank, you need to be a researcher at an institute that's recognised to do research, but also you need to formulate a research question and a proposal, and that then gets reviewed as part of their sort of before they would let you give access to the data. So definitely with all the approvals, with thousands of researchers, 25,000 researchers worldwide with approved applications, I definitely know developing and using UK Biobank and generating synthetic data sets from some of the UK Biobank to develop models is ongoing. So no question. And clearly UK Biobank has also got applications in from lots of different pharma companies who clearly are also leveraging the data. So I think it's totally is happening, but it'd be on an application by research question basis.
Right. Okay. And one last question before we go to the one in the Q&A. So you mentioned that maybe most of the patients in UK Biobank have kind of one-off samplings for biomarker analysis, and you do have a separate kind of set where they've been kind of followed up later on. But I guess even with the follow-up, is that still a one-off sample, or is there any kind of temporal tracking of biomarkers in UK Biobank? Because I guess the timing of sample collection might be important, and time of day.
Yeah. No, really a key question. So at baseline, everybody gave a sample. 20,000 people came back for repeat questionnaires and further samples. And then as time has progressed, as a research community, UK Biobank, what they're able to sort of enable comes from questions by the research community. So part of that research community has been, well, can we go back via questionnaire and get people to come back to give further samples, to answer further research questions? And a good example of that is the imaging study. By recontacting people, 60,000 people are going to come back for repeat imaging. And at that imaging study, it's going to be repeat questionnaire, samples taken as well. There's going to be proteomics, metabolomics on another 60 to 100,000 people. So, but you're absolutely right, at that particular point in time, they will just have it X number of years later. That varies in each individual, but then clearly your modeling can sort of account for these things.
Yeah. Okay. All right, interesting, thank you. So let's come to the question in the Q&A now, which I think is directed to Srijit. So this question is, do you think instead of completely stopping animal studies, that AI ML models would help researchers to set up maybe very specific animal studies to generate data and then rapidly translate that data into first in human studies. How do you see AI ML models helping there against the kind of push towards NAMs as well?
So there is work on decision making, and so this is a separate context, is to guide which animal studies to run. And there are approaches now that are working with different agents that interact with each other. They really like simulate the entire meeting that you would have in an investigative toxicology team, where somebody says, "Oh, welcome to the meeting. We want to discuss today about this compound causing this effect." And then another AI model is like, "Well, first of all, let's talk about the on-target activity data set that we have, and then let's talk about the metabolism. And we know this gene is overexpressed in this monkey, therefore we should choose something else as the animal model, and blah blah blah." So there are decision making work that happens, and I have been running some of those simulations as well. It's really fun, and to see how close to human decisions can those AI agents come for for an experiment that isn't public out there. So it's not published, or it's not out there for models to already know about. And some of the basic understandings it is able to understand and make reasoning out of. The problem comes when there are multiple different data sets to access. So you have to look at UniProt to find the proteins, and look at Campbell or internal data sets for the IC50 values, and look at the expression in animals, and look at the physiological map to figure out whether it's implicated in the process or the indication, and whether that might be causing the off target, whether it's the compound or the metabolite, blah blah blah. So there's like hundreds of different things. In theory, machine should be able to optimize for a lot number of parameters compared to humans. The models are just not optimized yet, but I do see a lot of companies kind of tackling this problem now, is to not replace humans in decision making, but to augment the efforts by being able to access or optimize a lot more parameters than humans can in one go. And by that, what I mean is, say, when you buy a house, you think of maybe 4 or 5 things at a time, and you spend 3 months thinking of 4 or 5 things each time, and then you end up thinking about 30 things before you buy the house. A model should be able to do those 30 things in 1 week. And that's what I mean, multi-parameter optimization, where the number of parameters is a lot more than what humans can do at a time.
All right, that's great. Thank you very much. I don't see any other questions. So we're maybe 4 minutes before we're due to break for lunch. So we could probably wrap up the discussion there, unless anything comes in very quickly. Oh, there is someone with a hand up. Joe, you have your hand up?
Yeah, sorry, I can't seem to ask a question. I tried to write it in the chat. Really interesting talk. I just wanted to ask, Sergi, what your feelings are. You mentioned a little bit about PK and PD parameters and the need for sort of curated data sets in those spaces. Do you have any thoughts on which databases are the most useful at the moment, and what's the real need in that space?
So for pharmacokinetic properties, there were standard data sets that were developed, I think, 5-10 years ago. Lombardo et al. Compiled a list of compounds, Cmax, and they have volume distribution and clearance and mean residence time and fraction unbound in plasma, all these endpoints. The problem is the variability across animals are quite huge, and therefore in this field, machine learning has come up with new metrics of analyzing these models, called twofold, threefold error, which if I were to tell you I can predict tomorrow's weather is going to be between 0 and 200 millimeters of rain, that sounds ridiculous, right? For a 50 millimeter rain, anyways. But in PK property prediction, you can go up to twofold error, and that would still be acceptable. Such difficult it is to predict these data sets. So there are private data sets that you can purchase for a price. There are companies that are curating these PK properties from patents and literature, and like really doing this by hand for the last 10 years. There are now AI models. A lot of startups will claim to have PK data for 30,000 molecules, and that's mostly coming from patents or from FDA filings, etc. That seem to be being more and more public now, which was not the case before. So there is more and more data coming in. In the public domain, if you are there, you're probably much more limited. If you are in a startup and you can, or if you have funds to pay for private access of these data sets, there's a lot more cleaner data with standardized workflows that you can access.
All right, great. So yeah, we're about on time now. So let's wrap up the discussion there. Thanks to Patricia and Srijit for sharing their expertise, and thank you to everyone that contributed to the discussion, those of you that posed questions. We're ready to start the invited oral communication session now. We have 3 presentations that were selected from the abstracts that we received late last year. We'll be awarding prizes for the oral communications at the end of the meeting. So please stick around for the closing remarks to find out who the recipients are. We'll run these presentations slightly differently, with each speaker having 15 minutes to present, followed by up to 5 minutes for questions. So again, if you think of any questions during the presentations, please post them using the Q&A feature, and we can try to get through as many of those as we can at the end of each presentation. All right, so let's begin with our first oral communication, which is from Zhonghui Wang, who's based at University College London. So Zhonghui, if you're ready, over to you.
Yes, sure. Okay, I'm gonna share my screen. Okay, so hope everything worked fine. Can you see the share screen and my sound? Hear my sound?
Yeah, that's great. Thank you.
Okay, perfect. Okay, let's start. Hey, so good afternoon, everyone. So it's a pleasure to be here to introduce our package developed by our group. It's just a message from University College London. And this package is designed for automated population pharmacokinetic modeling. Maybe some of you know pop PK modeling. It plays an important role in the drug development and clinical treatment. And this package try to utilize AI search algorithms to make the model selection ultimately available. So to help you to have a good understanding of it, I will start with some basic concepts of the population pharmacokinetics and and some general workflows for it. So for PK, so pharmacokinetics, it studies on the drug behaviors in human bodies. The process includes absorption, distribution, metabolism, and expression. So just like what the left figure show here, we usually get the drug concentration data. And the task is try to find mathematical models to describe the data. And then we can use that models to do the prediction, extrapolation, to support the drug development or treatment, personalized treatment. So we can propose some simple models or complement models. And here in the right panel, I just give examples for one complement model. We got the pod absorption part, and we got central volume distribution, and then we got the PK parameters to help us to capture the characteristics of the drug behavior. And then we get the model. What we expect is after modeling, what we expect is to get the predictions for population pharmacokinetics. We expect to get both population predictions and individual predictions. So the first one, population predictions, refers to over average trend of the population, and for individual prediction, like individual concentration profile for each subject. And then how we get these predictions So we use a model called nonlinear mixed effect model to help us to characterize the typical drug behavior and analyze the variability. And I post the general expressions here. And it mainly includes three parts. The first one, we call it the nonlinear function. So it's from the structural model, so describe the systematic part. And then second one, because we need to analyze the variability. So we introduce the IV model, inter-individual variability model at the parameter level. So to, analyze the difference between the individuals. And finally, we got the residual error, so which is commonly used in nonlinear regression, so which is to describe the differences between the observations and the predictions. And in the practice, so for a modeler, for a modeler, how we conduct the topic analysis. Currently, we have an open source modeling tool called Anomics R2. So, but still, modeler need to do some coding work and need to specify the parameter model and the structural model we like. And then after running, we can get the parameter estimates and also get the model predictions. As I marked here, the PRIDE and IPRIDE, which refer to the individual predictions and population prediction. The challenge here is actually we cannot use one model forever, to describe all of the data we get from the clinical trial. So for modelers, we need to think about, how to design each part for the models. And we have many options for each part. And after different decision, and after we use different models, we could get different model performance, just like the right dynamic figure show. In the right figure, the points shows the observed data, and the shadowed area shows the predictions. And you can see under different NLE models, the shadow area shows different shape, and some models can cover the data well, but some models doesn't have good performance. So it's a question how to select the appropriate model to describe data and then help us do more analyze the work for drug development or clinical treatment. So this means for modeler or for modeling, we really need risk experience, and then it's also a time-consuming work. It can take sometimes it can take weeks or even months to find a appropriate model. So that's why Anomics R2 Auto here is here. So we think we can automate all of the process ultimately. But this means now Anomics R2 can automate initial estimates, model building, evaluation, and we introduce some meta-heuristic algorithms into Anomics R2, so make it available to do automated model selection and optimization. So the goal here is we just need the user to provide Anomics R2 standard data. So we just need the data, and then these app packages can give the output the selected model. So we don't need to do more coding or manual selection work, make it more easier. And then how Anomics R2 do this? I will start with initial estimates. So currently, for Anomics R2 family, we have the first package, Anomics 2 Auto Innate, so which works on initial estimates. Initial estimates is a concept in parameter estimation. So when we try to estimate some parameters to fit the data, so we need to give some start values to let optimizers run. And good initial estimates are essential. If we give a poor initial estimates, it could make the model crash down. Our team developed the Anomics Auto Innate. So these packages use some data exploration-based analysis method to help to calculate the PK parameters and to give some recommendations on initial estimates. In the Anomics Auto family, This is the first package, and it is in the GitHub and in the R CRAN as well, so you can use it now. And then for model building, currently Anomics also cover 11 model components. And for the search space, it can test in theory, it can test more than 700 intravenous cases models and more than 1,400 oral models covered. And I also posted the details of the model components here for reference. For model evaluation, because if we want to select a problem model, we need to know which model is good, which model is bad. So Anomics are two auto covers several evaluation criteria. So it's mainly currently it's mainly divided to two parts: goodness of fit or parameter reliability. So this is finally Anomics auto will form a component matrix reflecting the two sides. What we can get from Minamoto is like what the right table show. After we run it, we can get the ACBAC or fitness calculated based on the matrix I mentioned. So this part is also automated, and then after we get the evaluation criteria, and the next thing is how we do the selection ultimately is related about search algorithms. Anomics also currently have 4 search algorithms, 3 meta-heuristic algorithms, and 1 step model building algorithms. We try to mimic what the modeler do. So the general workflow for Anomics Auto for each algorithm is like at beginning we will test some solutions, some models, and then evaluate those models, and then pass those models to different search operators. Different search operators have different way to update those models, optimize models, and repeat the procedures again and again. Finally, it can help us to convert to the best model. This is the general workflow. And then I will quickly go through some details of the search algorithms introduced into Anomics Auto. So for the first meta-heuristic algorithm, genetic algorithm, it was proposed in the last century. And Auto follows a previously published idea previously published the GA idea in the POPK. So currently we make the model as a chromosome. And then if you go through some key model update the procedures, including terminal selection, crossover, and the mutation. I will simplify this procedure and make it simply. I will make a simple explanation on this procedure. So what's going to happen in GA is, so at beginning we test some models and we get the performance of those models, and we will know which is good model, which is a bad model. And then for GA, what happens is the good model, the good model will share the higher probability to be selected for the next iteration. And after repeated procedures, finally we can get the model with the good model features. This is what happened in GA. And the next one is ant-colony optimization. I think this is the first time to propose ACO in the POPK analysis. So similar with the genetic algorithms, so ACO, it will also test some models at the beginning, but the difference is after we get the feedback of the models, we will allocate the probability on model features, not the whole models. And then when we test the next models, we will do the sampling work on each model features and finally combine a new model. So this is how it updates the model in ACO. So in Anomics Auto design, it is the SACO, simple ACO, and they use the rank-based update, which means we use the ranking of the model performance as the selection criteria. And it's also available to do some customized design. For example, for the first iteration, we can test the models we like. So we can choose some fixed route at beginning. Yeah, and the stop criteria for GA and ACO in Enormous Auto is the number of the iterations we like to test. This is GA and ACO, and the third meta-heuristic algorithm is the table search. This is kind of like a local search. So what happens in this algorithm is that it will test the one model at the beginning, and based on this model, we will test all of the neighborhoods around this model and pick up the best one. And in the neighborhoods, after we pick up the best one, and based on the best model, and also test all of the neighborhoods as well, and then repeat the procedures. But the only thing, special thing happen in the table search is we have a memory list. So we will write out some model features that we have already tested. So in the next iteration, we won't test those features. So that's how it works in table search. And the end result also provide a stepwise model building algorithms. These one try to follow a fixed order to test the model features. As I list here, currently it starts from structural model, followed by IFV model, ending with residual error model. But Enormous Auto also allow you to, design your own order. It's no problem. And this model building workflow is trying to mimic what human modeler do. And then here it is Enormous Auto overview. So what happened is, so we need users to provide Enormous true standard dataset, just this dataset. And then this dataset will be passed to Enormous true auto innate, and this package will handling the data and then calculate the basic information to PK parameters and provide some appropriate initial estimates, and pass these initial estimates to Enormous Auto, which include the search algorithms inside it. And you can specify any search algorithms you like, and run these algorithms. We can get the selected models. So after we design the package, we also do the test work for these algorithms. We want to know, the performance of the algorithms. So we create 144 simulated datasets to test it. And besides, we also collect the 22 published data from the publications. So these datasets cast various of the PK scenarios, different sampling design, or different dosing regimen, different population, and different sample size. And the right figure here is the 22 is the drug concentration profiles for the 22 real-world data. And how we evaluate the algorithm's performance. So now we have the search space, so which means includes all of the candidate models. And then we conduct the exhaustive search to get the best model. So this means we test all of the candidate solutions and then pick up the best one to treat it as the ground truth model. And we calculate the percentage numbers of the cases by using different search algorithms to match the ground truth model, identify the exhaustive search. At the same time, we also calculate to record the runtime for the search algorithm. And this here, we get the results. So Enormous Auto have 3 meta-heuristic algorithms. And it's good that we see, all 3 meta-heuristic algorithms performs good. The accuracy rate, close to the 90%. And in expectation, the stepwise model building algorithms perform markedly lower. These algorithms try to mimic human modeling procedure, but currently we found that the accuracy rate is low. this is the results from simulation 144 simulation study, and and then we also record the runtime. So this from the results, we found that for per each data set, it needs about 4 or 5 hours to run a data. And the average unique models number, the average unique number models are about 100 or 120. So it's corresponds to like 15 or 17% of the search space. For real-world data, we also found a really good performance for the 3 beta-accuracy algorithms. And they just missed 1 to 3 cases when we compare with the exhaustive search. And similarly, the stepwise procedures and performed worse than the other 3. And for real-world data runtime, honestly, it is varied. But more than half of them can finish in 1 day. So it is also quick as well. So the conclusion here is we designed the NOS auto, and it's worthwhile. And then after we test the algorithms by using simulated data or real-world data we collected, it's also have a good performance. It also tells us that the AI search algorithms enable automated model selection and optimization. It's an open source tool. So this means it's lower the barrier to the POPK modeling, which means if you are a researcher from like a hospital or academia or other fields, if not quite familiar with POPK model, I mean, this is a good tool to help you to have access this technology and help you to do the modeling work and utilize it in the drug development and treatment. The future work for Anonymous Auto is about covariate models. So it's focused on the base model development, and we plan to add a covariate model part, and then after the covariate model, we will have a look at how to utilize the search algorithms in the pharmacodynamic model selection. Yes, that's all of the presentations, and thank you for your listening.
All right, great, thank you. I don't see any questions in the Q&A, so maybe we've time for one question before we move on. So it seemed to me as though this was all based on kind of clinical human PK predictions. How does the availability of the actual real world data impact your ability to kind of validate that model? Is there any opportunity to use in vivo data as well to boost your ability to validate the predictions, or are you really reliant on the clinical studies having been performed already?
Sorry, I mean, whether this is the data-driven algorithms, is it algorithm the question? So Anonymous Auto use the search algorithms. So which means as long as we finish the algorithm design, it is really flexible. So it's quite easy to apply this data set to other clinical data. So I mean, we don't need to train too much clinical data. It's not like a machine learning, just we need to have the training data set and do the more extrapolation. It's a search algorithm. As long as we test some data and get a hyperparameter of those search algorithms, and then we can apply it on other data set. Hope it makes sense.
Great. All right. Thank you very much.
Thank you.
So let's move on then to our second oral communication. So this is from Mark Ellison, who's based at INSTEM. So Mark, if you're there and ready to present, over to you. Thank you.
Okay, can you hear me okay? And can you now see my screen? It should be a slide.
Yeah, that's great.
Okay, great. So today I'm going to be talking about when Send meets AI, where it works, where it breaks, and why that matters. So for anybody who's unfamiliar with the Send standard, hasn't had the pleasure of working with Send datasets yet, Send is the industry standard for a machine-readable electronic representation of our non-clinical in vivo toxicology data. And it's also the required format for FDA submission of those data at IND and NDA. In fact, the FDA making Send a regulatory requirement back in 2016, that's led to the wide adoption of the Send standard across the industry. So in the context of AI working with Send datasets, there's two sides that I'm going to be mentioning briefly. Firstly, using AI to assist in rendering our study data into Send. In other words, assisting in the standardization process, what we call generating the Send datasets. And secondly, in consuming the large volumes of Send data that now exist in organizations and organizations have access to.
Mark, sorry to interrupt.
Yeah.
We can only still see your first slide, is that right?
Yep.
Okay.
So you should be able to see my second one now.
Yeah, I didn't know if you meaning to announce that it hadn't worked.
That was me. That was me simply explaining the title.
No, that's great.
Okay. So this is me. I've been a CDISC volunteer for almost 15 years now. CDISC is the organization that creates and maintains the Send standard. And so I've been helping to define that non-clinical standard for, I'd say, about 15 years now. My early career focused on non-clinical data capture systems. But for the past decade or so, I've been focused on representing data in Send and then exploring the possibilities of reusing those data once they're in a standardized format. So this presentation is coming from my personal point of view of using AI with Send kind of as a consumer. So we'll use AI to a greater or lesser extent now. And so as kind of as an introduction, I want to start by telling my story of using AI in a personal healthcare situation. Back in June 2025, last year, I got COVID, which left me with a severe laryngitis. And I lost my voice, and I ended up losing my voice for just over a month. And during that time, there was 3 weeks where I was having tests and things where it genuinely looked like I'd never be able to talk again. As you can tell, talking is kind of important to me. So what I did was I created an AI model of my voice, and because of what I do, there's plenty of training material available, so I could give it recordings, and I trained an AI model of my voice. And the results were actually incredibly convincing. As you can tell, I did get my voice back, but even colleagues that I've worked with for nearly a decade now, they were completely fooled by the results and couldn't tell the difference between the AI me and the real me. So another example of using AI in everyday life, we're all familiar with ChatGPT. You may be familiar with the strawberry test. We asked ChatGPT how many R's are in the word strawberry, and it tells us that there's only 2, and we can all clearly see that there's 3. So while there's different types of AI, for today's short presentation, I'm going to look at just 2. The impression of my voice is a good example of generative AI, while the strawberry test is an example of reactive AI. And generative AI is good for performing specific tasks, such as replicating my voice. And the reason a strawberry test fails, and is so infamous, is that the AI is really good at predicting. I'm going to do AI a huge disservice now and oversimplify things. But for the purposes of today's discussion, I'm going to think of AI as a really good guessing machine. So when we ask the question, how many R's are in a certain word, 2 is probably a pretty good guess, and it will be correct quite often. So the key point here is that unlike a calculator or other computerized systems that we're used to using, AI doesn't always give us a calculated answer, but rather makes a very good and often very convincing guess. So when representing data in the SEND standard, this can be really time consuming, can be quite labor intensive. And one of the challenges is ensuring that all of the terms used, like units of measurement, tissue names, test names, those kind of things, well, we have to make sure that they use what we call control terminology. So what do I mean by this? Well, in SEND, we need to make sure that we all use the same terminology. So if one organization calls the organs the adrenals and the other calls the organ the adrenal glands, we all need to standardize on the same term. And in SEND, that is gland, adrenal in uppercase. So clearly, AI can be a great assistant with standardizing non-clinical safety data and applying the appropriate standardized terminology. It can be great at identifying terms that need standardization, but we still need the human in the loop, as the suggestions that can be made, well, they can be typically very good, but they're not always exactly right. And this is how I use AI when standardizing data. It's a great assistant, but it's not a replacement for the subject matter expert. Recently, I used Copilot to assist me in applying control terminology. It identified terms to be standardized, and it made some great suggestions. But I'd say of these suggestions, it was probably only about 70% appropriate. The other 30%, it either couldn't make a suggestion or suggested something that was obviously incorrect. The most extreme example is when there was a measurement for temperature where it didn't recognize the collected unit of C. And so it suggested that seconds might be the appropriate unit of measurement there. So obviously that was incorrect. So it's always, that's why we always need to have the human in the loop to catch these things. And that's because when dealing with the submission of safety data for first in human, AI's best guess isn't good enough. The SEND data sets need to be 100% accurate representation of the GLP study data. And we also need to consider that with safety data, they need to use validated systems, so that we need to know that they produce consistent and predictable output. And that obviously there's concerns about data privacy and use too. So there are many opportunities for generative AI use in our industry. On this slide, due to time constraints, I'm just going to talk about the box on the right. So regarding the use of SEND data, AI can be great for making complex searches of data and drawing conclusions. Performing tasks are too complex for traditional database searches. So I'm thinking of instances like where we might want to look across many studies and find instances where the compound caused a decrease in body weight combined with maybe significant findings against certain tissues, and maybe combining that with significant changes to certain clinical pathology parameters. One of the great features of SEND data is that while the data are designed to be machine readable, they're actually pretty human readable too. I mean, if we cracked open a body weight domain, for example, SEND data, we'd see things that we recognize. We'd see the word grams, we'd see phrases like study day 1, we'd see things that we recognize like dates and times and subject identifiers and the like. And it actually makes SEND pretty human readable, even though it's predominantly designed to be machine readable. So this means that we can easily detect any AI hallucinations when it comes to drawing conclusions, as we can simply ask the AI to show us the records that support the certain conclusion. And as the data are virtually human readable, it's actually quite easy to then check the results for ourselves. Okay, so now what about reactive AI? So reactive AI is great for deterministic, repetitive QC tasks, but it's not sufficiently nuanced for compliance decisions. It's best seen as a component of a hybrid approach where reactive AI handles rule-based validation and automation, and human experts handle interpretation and judgment. And so this is why I'm continually referring to needing the human in the loop. So there's many scenarios where AI may not be appropriate. Now, due to time constraints, I'm not going to talk through every point on this slide. I'm actually just going to talk to the final point here on the right-hand side. I've now seen several organizations that throw data extracts or even final study reports at ChatGPT, Copilot, and the like, and ask it to generate the appropriate SEND data sets. And I'll say that what is produced is really impressive, but it's not totally correct or compliant. But unless we're well versed in SEND, we could easily be fooled into thinking that the results are acceptable. As I said, from pocket calculators to Google searches, we've grown up with using IT systems and computer programs with complex algorithms that always produce the correct results in a repeatable and predictable way. But when it comes to using AI in this way, we need a mindset shift. We can't simply take the results on face value. We shouldn't reduce the human oversight. And it's easy to have overconfidence in AI, but I always think back to remembering the strawberry test that I mentioned earlier. So here's my version of the strawberry test. We asked AI for a definition of element in SEND. Element is a particular variable in SEND. And the AI had already read the SEND standard. It could quote it. However, when we asked it for the definition of element, it got it completely incorrect. Copilot told us that the element is a single piece of data and gave us examples such as a subject ID or a dose amount or a observation result. But actually in SEND, element is a planned period of time in a study. So pre-treatment would be an element, for example. Daily dose of 100 milligrams per kilogram would be an element. Recovery would be, etc. So these are all three elements that you typically find in repeat dose study with a recovery period. It's a planned period of time in a study. So Copilot gave us a completely incorrect definition, yet it did it so convincingly that if we're not well versed in SEND, we'd simply believe it. Oh, sorry, jumped ahead too far. So before I finish, I want to make one final point. We've all seen news reports of when AI advice or information is incorrect. Whenever we use ChatGPT, Copilot, or the like, we'll see some disclaimer stating something along the lines of AI-generated material may be incorrect. I'm sure we're all familiar with seeing those disclaimers on screen. I want to contrast this with the statement in the FDA's Technical Conformance Guide. And this is specifically referring to SEND datasets. Now, it says that these data form the basis for the rationale on how a sponsor concluded that it is reasonably safe to conduct the proposed clinical trial. In another part of the same document, it talks about the FDA needing to ensure that the SEND datasets are fully traceable back to the GLP study data. So the agency are making their decisions about the safety of the experimental new drug based on the SEND data, and they're trusting that the SEND datasets are an accurate representation of the study data. So, in conclusion, my final thoughts. AI is not an expert in SEND or safety data, but we could easily be fooled into thinking that it is. AI should be like having a great assistant that helps us. When we use AI, any safety decisions and any safety data need to be traceable and defendable. So AI offers us a great opportunity to improve efficiency and to be able to handle large volumes of data and assist in pharmaceutical R&D, but only when we keep the human in the loop, we ensure that all the results and conclusions are checked and that the data are trustworthy and traceable back to the original study. And with that, I will thank you for listening. I've been Mark, and until next time, I hope you found this interesting.
All right, great. Thanks, Mark. I don't see any questions in the Q&A, so maybe I could just follow up on that point you made a couple of times about the importance of the human in the loop. So I kind of asked a similar question earlier to one of the speakers, but do you see a point in the future where these models or predictions will be so, we have so much confidence in them that the human in the loop won't be required, or maybe it'll be required to a lesser extent than it is now? What's your feeling on how things will evolve in that way?
I think things will evolve and improve. And I mentioned just that it got around, when I did the simple test, it got around 70%, made 70% accurate suggestions. Absolutely, we'll see that increase and get better. I'm sure we'll quickly get to the day when Copilot doesn't suggest that seconds will be an appropriate unit of measurement for a temperature measurement. Will it ever get 100% right? I don't know. Would we be able to trust it less? Yes, probably, but still, there would. Could we use AI to help test the results that AI generated, for example? Yes, you could do those things. I think it will improve. I don't think it'll ever get 100% there, but just in my own use of it, just the difference in between ChatGPT 4 and 5 was huge and really shocked me when I first saw Send generated from ChatGPT 5. So it'll get better, and the rate that it's accelerating is very surprising, but we still need that human in the loop. One of the things that I didn't mention, simply because of the time, when a human checks something and a human puts their name to it and says, I have checked this, this is good, this isn't 100% accurate representation of my study data, that carries more weight, or that carries some weight. Somebody is investing something of themselves in making that decision and making that declaration. If we trust an AI to do it, the AI doesn't have to. And there is debate about, well, is that good or bad? Does that remove bias? There's those kind of things as well. Personally, maybe I'm just old school. I still like a human being to take responsibility and say, yes, I've checked these data. These data can be trusted. I don't know if I answered the question or not, or if I just wandered onto a different topic.
No, you did. That's great. Thank you very much. All right, let's move on then. So thank you to Mark. Our final oral communication is from Joe Standing, who's based at University College London. So Joe, I see you're on the screen. If you're ready to share your presentation, please take it away. Thank you.
So are you able to see my slides?
Yeah, that's great.
Okay, well, thank you very much for the introduction and for the invitation to speak. So I'm Joe Standing. I'm a professor of pharmacometrics at University College London. So I'm primarily a pharmacologist. And the work that I'm presenting is in collaboration with the main person who's done the work is Vicky Smith, who's a PhD student in our group who finished earlier this year. Ferran, who is a former PhD student, a colleague of ours, Watty, who's from UCL Computer Science, and Frank, who's another pharmacologist. And we have developed something called PKPD AI, and I'm going to present a little bit about what we've done in this talk. So as background, we've heard about what pharmacokinetics is in previous talks in this session, and we now know that in order to describe pharmacokinetics, we need to fit these nonlinear models, which may be simple or they may be quite complicated. So this is an example of a physiologically based pharmacokinetic model that's got lots of these nonlinear parameters that might appear in the literature. And around 2018, so sort of 7 or 8 years ago now, I had this idea. Well, I typed in pharmacokinetics into PubMed, and there were about 600 to 700,000 papers. So pharmacokinetics, we started measuring drugs in about the 1960s. So, it's sort of a relatively mature discipline now. And, there's too many papers to read. So the idea I had was, can we teach an algorithm to read the papers and then extract the parameters and do some of that for us? So I went to speak to people at computer science, and Watty, who's our collaborator, has helped hugely with this. And this is sort of my vision, is that we've got in a peer review publication, we've got some text, and this is pre-published work, so you can read about our text mining. But what I'm going to talk to you today about is how we then also have tables. And within that table, firstly, you need to decide, is this a relevant table? Does it contain useful PK information? And then once it does, you have to decide, well, does it have interesting and useful information in it? So does it name the drug? Does it have a parameter? Does that parameter have units? Do those parameter and units link with a numerical value? does that numerical value have some degree of uncertainty? So you need to recognize these entities. You then need to standardize them. So we call AUC 0 to infinity in this example can be called lots of different things. This infinity symbol could be replaced with inf or something like that. So we want to standardize and normalize the entities. We then want to establish relationships between them. So to make sure that an algorithm can understand that this parameter is linked with this numerical value, and this plus or minus is some measure of dispersion, and this is its units. And finally, we want to put it all into a nice table, because wouldn't it be lovely if we had a table of all of the drugs, of all of their parameters, of all of their values and their deviation values and their uncertainties and their units and the root in a massive, massive data set? Because if we did, we could start to do machine learning to predict, from known chemical entities, what was their pharmacokinetics, and can we now predict a new chemical, what's its pharmacokinetics? Or if we know the pharmacokinetics of a drug in the dog, can we predict it in the human or the mouse or the child or the preterm neonate or the elderly person or someone with renal failure? So this is a sort of vision. And there are these databases around. I think some of them were mentioned in one of the earlier talks. But the problem with a lot of these databases, the vast majority of them, is that they're human curated. So a human has had to take the time to extract the data and put them into a database. Some of them are behind paywalls. And for the vast majority of them, there is only a single entry for each drug. So we have perhaps the human healthy volunteer clearance of a drug, whereas we might be interested in all of the human data from all different patient types, or the dog data, or the mouse data, or the fish data. So what we thought is, if we can automate the extraction of this from published papers, then we could generate data sets at much larger scale. So in order to focus on this task of taking PK parameters from tables, we've got these 4 main tasks that I'll go through in the talk and show you how we've done it. So first of all, we have got to classify the tables. We've got to extract tables from PubMed Open Access. This was our corpus, but we could use others. And decide in the first case, does this table contain PK relevant information? Yes, no. And that's just quite a simple classification task. We then need to do named entity recognition to find out the types of entries in the table. We then need to do entity linking to standardize the names of those entities using something called a PK ontology. And we've used a pre-published one. I'll show you a bit about that. And finally, we want to link everything together and put it into a lovely table that will be ready for future AI and machine learning applications. So let's start with table classification. This task involves distinguishing the tables that contain in vivo PK data. When I say in vivo, I mean any species. So we're agnostic to humans or animals. And, well, we've written patient demographics here, but actually it could be animal demographics. What was the weights? What were the ages and things like this? And this acts as a filtering step so that we don't have to tell the algorithm, here's some tables that definitely contain PK, and then a human has to give them the tables. No, we can just go through all the open access tables in the whole of PubMed and give the tables to the next stage of the algorithm. So this allows us to be accurate, giving the future bits of the algorithm the right kind of data to work with, and scalable, because a human's not involved in choosing which table is relevant or not relevant. So we did this by using, we heard a question earlier about people in low and middle income countries being used as annotators. We use our students. So we had lots of help from PhD students, master students who were rewarded with things like acknowledgements and authorships rather than financially, but they were very keen and they were crucially subject matter experts. So we passed the tables into an interface called Prodigy. And what this does is if you've got the table in an XML format, you can put it into a really nice interface that you can see in the bottom left here. And then they can just look at tables very quickly and say, is this a pharmacokinetic table? Is it a demographics table? Or is it another? Classify it, then click done and go on to the next one. So we annotated a few thousand tables and managed to find an algorithm using a fairly simple classifier to have a good F1 score. Now, this classifier was called XGBoost, and it worked relatively well. But what we found is, so if it's really obviously a PK table or obviously a demographics table, then it works really well. As the F1 score goes down, as the confidence of the XGBoost algorithm goes down, so the F1 score decreases. And what we did there is we took the ones where the classifier was less confident, and then we fed it to a large language model. So in this case, it was GPT-4, and this massively improved performance. So we use the F1 score, and you can see that we've got F1 scores, which is a way to balance between false positives and false negatives. And you can see, and 100% means that we've got everything right. And you can see that we've got really high F1 scores. And this is a real testament to the fact that we've got very high quality labeled data sets with two people labeling the same thing. If they disagree, a third person comes in and makes sure that our labeled data, even if it's not huge, is very high quality. So we've now got an algorithm that can tell you what type of table it is. We then need to look at how to distinguish the parameters within the tables. And this is really difficult because you can see these examples of tables being with really different orientation. So the parameters can appear almost anywhere. Cells featuring the PK parameter spans might reflect broader concepts like parameter covariate effects rather than just the parameters, and variability terms. So we split this into two tasks. Firstly, can you classify the cell in the table and say if it's got what type of PK terms it got in it? And then can you link the cells with span named entity recognition in order to link, okay, well, here's the Cmax, for example, and then here's the numerical value that needs to go with it. And again, as you might imagine, if you're not a PK modeler, this was a really important task to have specialist people who understood PK modeling in order to be able to do this labeling. Once again, we fed it into a Prodigy interface and always had two annotators and an independent reviewer. And we looked at PK cells, other cells, and then total cells, did a 60-20-20 split of training, test, and development, and had a very large data set that was really well labeled. We then needed to pass this cell text into a BERT model in order to classify it. And you can see that our F1 scores were very high again. So we were able to categorize the entries in each of the tables. And yeah, this shows that we've, sorry, that's the same slide. So now on to entity linking. So entity linking is where we've got the, something might say AUC 0 to infinity or AUC 0 to inf, or, synonyms for the same thing. So this used a rule-based approach, and Wu et al. Several years ago published a pharmacokinetic ontology We've extended that, and on PKPD.ai, our website, which I'll refer you to at the end, we've published that ontology as well, so anyone is free to use it. So we've got more than 20 different parameter types. Each of them has got standardized descriptions and standardized units in terms of, mass times divide by volume. So it doesn't specify it has to be in hours or milligrams, but just types of the units. And we have logical categories to group the parameter types. And this allows us to do quite simplistic rule-based approaches to categorizing what the parameters are. Once again, common theme, we put this into Prodigy, and Human labeled it, had a really nice big data set again of well over 1,000 tables, and similarly sentences, so things from the legends and the titles. And again, we use this sort of funnel-based approach where we had we had the rule-based approach to do the exact parameter matching from the ontology. We then had some labeling, and then finally in the edge cases, we used the large language models to really improve the predictions. And I think, just to note, I think this is what we believe is sort of a responsible use of AI, rather than, instead of throwing these large language models at very easy tasks, we use simpler rule-based and then classification-based, and then in the edge cases, we use the large language model to really drive up our performance on our F1 scores. And again, we're approaching well over 90% for everything there. So we then wanted to output this into a database. So what we did, so then we've got to link the entity. So we've done entity linking and used something called Burn, which is a generic entity named entity recognizer that has helped to categorize things that are not in our PK specific data set. So for example, drug names and creatinines and covariates and things like that. So, and then we validated the database. So we've run it over some unseen data, and then human labeled 50 random PK tables and looked at how it performs on unseen data. So we were relatively happy with this sort of completely unseen data, and we then ran it over the whole of PubMed Open Access. So what we've got is we've found 177,000 PK parameters in PubMed Open Access, and the vast majority of those we were able to link the parameter with its measurement, with its units, with some measure of variability. What we were less successful in is linking that with the covariates, like the doses, route of administration, and in particular, for example, weight or age. And these are really actually going to be important covariates if we want to look at sort of predicting to different subpopulations. And adding the other covariates that we could get from the standard named entity recognizer, such as the drugs, the diseases, and the species. Again, this was variable. In the vast majority of them, we were able to capture the drug name, but to see what disease it was being tested for and what species it was being used in was less well captured. So these are areas that we'll be working on in the future. So in conclusion, we've developed this end-to-end, and it's a PK domain-aware natural language processing pipeline that has classifiers, named entity recognition, entity linking, and structured extraction, which is really supported by expert curated data sets. We've shown the value of combining traditional natural language processing, which, as I say, we started in 2018 before we'd really even heard of large language models. But then as the large language models have come in, rather than trying to replace our pipeline with LLMs, we've used them selectively where the simple approach doesn't work very well. And then we've demonstrated the feasibility and the practicality of automated table mining in the PK domain to lay the groundwork for future unified documents to understand systems. So in the future, we want to link further within the document. So we've done a lot of work on text mining, and we've now finished this work on table mining. We want to merge these together and see if that's going to improve our, for example, capture of covariates like the body weight and the species. We want to expand the coverage. So we've only made publicly accessible things that are publicly accessible already. So we've only run this over PubMed Open Access. There, of course, are all the closed access documents. There are PDF publications. We have a PhD student looking at clinical trial documentation at the moment and regulatory reports. And looking at this out of distribution detection and flagging novel or suspicious PK parameters for human review is really important, because as a journal editor, I can tell you things get by, unfortunately, even for the journal which I'm an editor of, and we find mistakes in published papers. And we need to acknowledge that they're, just because it's in a peer-reviewed publication doesn't necessarily mean it's correct. So I would like to finish by thanking you for your attention. The first iteration of PKPDI was funded by a Wellcome Trust Open Research Award, and Vicky's PhD studentship was from the EPSRC, so that's where we've got most of our funding for. But most of all, as I say, it's an open science award, and we have tried to make all this openly available. So please do visit PKPDAI, where you can try to things like finding papers that contain PK parameters, looking at our PK ontology, and also looking at the parameters we've extracted. One of the things that we're doing at the moment is currently automating all of this work, so that now hopefully in the next month or two, every 2 weeks, whenever PubMed is updated, the algorithm will run over it and the database will automatically be updated, because this is a huge challenge. There's something like, I think I checked last year, 25,000 PK papers are being published every year on PubMed. Of course, not all of those contain PK parameters, but what we want to do is keep our database up to date, and especially, bringing in PK of more novel, monoclonal antibodies, PROTACs, and all the new type of therapeutic entities that are coming in, all of which we're going to need to understand what concentration is reached in vivo to understand if these drugs are ever going to work. So thank you for your attention, and I'm ready to take questions.
All right, thanks a lot, Joe. There are 2 questions in the Q&A. I think we've probably only got time for one, because we're due to break now for a bit. So I'll ask the first one. I don't know, Joe, if you can actually see the Q&A. Maybe you're able to respond offline during the break to the second question. But yeah. The first one is, how good is PKPDAI extracting from NDA and scanned PDFs, which currently AI struggles to do those types of extractions?
Yeah, well, we also struggle, and we've got a PhD student who's looking at exactly this. So yeah, it's tricky. PKPDI pipeline needs the XML document, needs a conversion to XML. So that's what we're working on. For more modern PDFs, this is not such a challenge, but for older PDFs, it's very difficult. So that's a sort of a generic challenge that I think is common to a lot of data extraction pipelines.
Yeah. Okay, great. So yeah, as I say, Joe, if as a panelist you're able to maybe type an answer to the second question in the Q&A during the break, that'd be fantastic. But I think that just leaves us to wrap up the oral communication session. Thank you to our 3 speakers. Wish you all the best as you continue your interesting work, and thank you to those that have submitted questions and contributed to the discussion. So
to introduce myself, I'm Professor Janet Mifsud, and I'm from the Department of Clinical Pharmacology and Therapeutics in the University of Malta, and I'm also a fellow of the British Pharmacological Society. And it's my second year being on the organizing committee for this very interesting meeting together with the British Pharmacological Society. And I might take the opportunity to thank my colleagues of the ISER organizing committee, together with the staff both at BTS and BPS, for the excellent preparation in organizing all this meeting. So this afternoon session will be going on to look at some more practical aspects of the use of AI in pharmacology and toxicology, which is the clinical and regulatory aspect. And our first speaker would be Professor Alejandro Frangi from the University of Manchester. Professor Frangi is a bio-CNT returning chair in computational medicine, University of Manchester, with joint appointments both in computer science and health sciences, so very interdisciplinary. He holds the Royal Academy for Engineering Chair in Emerging Technologies and leads the UK Centre on in Silico Regulatory Science and Innovation UK. And his research integrates computational modeling, simulation, and AI, while enhancing safety in healthcare technologies and drug development. So really bringing all the key disciplines in this area together. Unfortunately, Professor Frangi could not be with us in person today, but he sent us over his presentation, his recorded presentation, and I'll ask our colleagues to start the presentation going. Please, anyway, if you have any questions for him, we will forward them to him afterwards, so please don't hesitate to write them in the chat. So over to Professor Frangi. Thank you.
Hello everyone, sorry I couldn't be online today for this talk. My name is Ali Sarami and I'm a lecturer in cardiovascular biomechanics at the University of Manchester. I'm here today on behalf of Professor Alex Frangi to give you an overview of the UK Centre for In Silico Regulatory Science and Innovation, or the UK CERSI. I'll share our vision and explain how we are advancing the credibility of in silico trials as a new complementary source of evidence for risk assessment and regulatory decision-making around medical products, including medical devices and pharmaceutical products. Before I start, a quick disclaimer: most of the slides I'm using today were prepared by Professor Frangi, so all credits goes to him for the content. One of the oldest principles in medicine is the Hippocratic Oath, which says, "First, do no harm." The principle is directly relevant to the theme of this event, risk assessment. There are still important challenges in how we currently generate evidence for safety and efficacy that sometimes can put this basic rule at risk. And at UK CERSI, our view is that in the digital era, when Hippocrates meets Turing, do no harm should be complemented by simulate first, using AI and computational simulations to explore the risks first before exposing the patients. For every medical product, no matter if it is a drug or a medical device or even a combination therapy, we must generate evidence for safety and efficacy through the development and regulatory life cycle of the product. And traditionally, we've been relying on bench testing, animal testing, and human testing. These are all well-established approaches and supported by decades of scientific practice, but they have also their known limitations. Multiple studies show that despite these well-established frameworks, patients are still experiencing significant harm. In the US, failures in just 10 key medical devices over a decade were linked to around 83,000 deaths and around 1.7 million injuries. And this is not just the US. The compiled report, First Do No Harm, highlights very similar issues in the UK as well. Most recently, we saw the withdrawal of the last remaining left ventricular assist device from the market after multiple recalls. And when you look at the FDA recall data, just in the first half of 2024, we see recalls affecting almost every major company in the sector. So this is not a problem in isolation. This is a systematic challenge, we think. An analysis by colleagues from FDA and ANSYS showed that for every 100 complex medical devices entering the clinical testing, only about 30 of them will receive approval from FDA, which means that around 40% of the patients enrolled in those clinical trials are exposed to investigational devices that never reach the market. In pharmaceuticals, it's even worse, with 5 drugs out of 100 succeed, meaning that the vast majority of the trial participants are exposed to products that will never be approved, which means a large number of patients are facing real risks without any clear benefit. Another issue is time. It takes around 12 years to develop a new drug and around 7 years for a medical device to be developed. Clinical trials can be very expensive, with around, for a complex medical device, it typically costs around £50 million if everything goes right. And it can be closer to £500 million if we include indirect costs and the failures. Patient recruitment is another major bottleneck, with around 80% of trials being delayed just because of these patient recruitment problems, representativeness problems, and also the dropout rates are the big challenges. So basically, we are working with a system which is a slow, expensive, and sometimes inefficient. And at UK CERSI, we are interested in reducing the development timelines from years to months and failing earlier rather than later in the product life cycle and acceleration of safer innovation. Our proposition is basically very simple, with simulation first. When we say in silico trials, what we mean by in silico trials is basically computer-based clinical trials. We mean we start from virtual populations of patients and we simulate the interventions and try to understand what is the safety and efficacy rate for those devices that we simulate. And once again, we are not advocating the elimination of bench or animal or human testing. We are actually thinking about a fourth route of evidence generation, which is modeling and simulation. And looking forward, we see a hybrid evidence framework that can bring the failures earlier in the product life cycle and make them cheaper. The main components of in silico trials, as I mentioned, the first component is generation of a virtual population of patient anatomy and physiology. Then we need to mathematically model and simulate the examinations and the interventions, and we need models to be able to predict the clinical outcomes. These are the three main components of this in silico trial space. To give you a few examples, one of the in silico trials that we did in the past, the main idea was to see how reliable are these in silico trials. This is focused on flow diverter devices for treatment of brain aneurysms, which are pathological balloonings of the brain vessels that can rupture and put the patient in risk of severe complications. And one way to treat them is to put the flow diverter in the parent artery at the neck of the aneurysm to block blood flow from flowing inside the aneurysm. And then a clot will start forming inside the aneurysm sac, and this aneurysm will be isolated from the circulation. So the main idea is to reduce blood flow in the aneurysm. The clinical trial that was used to get the approval, the FDA approval for this device, was the PUFS clinical trial, which took around 8 years, and it was quite expensive. So what we did was we tried to generate a virtual population. We started from a reference population of patient anatomies based on angiographic images. We applied the same inclusion-exclusion criteria as the pivotal trial to end up with a population which is representative of the real population. We compared different aspects of these two populations to make sure about the representativeness. And then we simulate the deployment of the device and the flow pattern before and after device deployment. And what we saw was that we were able to replicate the success rate as it was predicted in the pivotal trial. So what we got from this in silico trial was very, very similar to the real clinical trials. And we were able to do also some more comprehensive subgroup analysis and identify some of the risk factors for unsuccessful deployment, unsuccessful treatment, basically. which is the aneurysm size, patient being hypertensive, and presence of a branch artery emerging from the aneurysm sac. Another example is from the pharma, and this one, basically some of our collaborators in the UK CERSI have been using physiologically based pharmacokinetic modeling, or PBPK modeling, to simulate drug exposure in adult and pediatric virtual populations. The key point was simple. Drug behavior, including drug-drug interaction, can be very different in children compared to adults, and rather than testing every scenario in real patients, these models allow simulations to explore dosing and interaction risk first. In this case, the simulations were accepted by the regulators and used to support the pediatric labeling. So this is a concrete example of in silico trials being used in pharma to reduce uncertainty, limit patient exposure, and support regulatory decisions. Our main objectives in the UK CERSI is to understand how in silico evidence could be integrated into existing and emerging regulatory pathways across devices and pharmaceuticals, and at the same time be focused on scaling adoption by co-creating credibility frameworks and best practices that regulators and industry could actually use. We built a large and interdisciplinary community from regulators, industry, and academia, and we created 10 regulatory airlock pilots, 10 use cases from different medical devices and pharmaceutical products, and in each of them we created a safe space for regulators, industrial partners, and partners from academia to be able to talk to each other and discuss the credibility of in silico trials to be used as part of the regulatory decision making about these devices. We've been in close collaboration with regulators like MHRA and FDA, and what we hope as the main outcome is a position paper from UK CERSI that basically can provide practical recommendations on the credibility, evaluation, and adoption of in silico trials in medical product regulation. So to conclude, medical product innovation and regulation are clearly at an inflection point. Computational modeling and simulation and AI are no longer experimental. They are becoming an essential ingredient, already well positioned within the regulatory pathways. This is not just a technical shift, but basically a cultural one as well. And this transformation is already happening, and its success depends on sustained engagement across regulators, industry, and academia, with both regulatory pull and industrial push. If we get this right, Hippocrates meeting Turing becomes not just a metaphor, but a practical route for safer and a smarter innovation. Thank you for your attention, and these are basically the ways that you can contact us at UK CERSI. Thank you.
Thank you very much, Dr. Ali, who was speaking there on behalf of Professor Aleandro Frangi. Extremely interesting presentation. Unfortunately, they couldn't not be live in person with us today. That was a recording, but you have their contact details, so if you wish for further information or clarification, please don't hesitate to contact them. So next in this afternoon session, looking at AI-driven innovation for smarter and safer drug development, it's my great pleasure to introduce Dr. Megan Howling. Dr. Howling holds an MSc in Biomedical Sciences from Radboud University in the Netherlands. She has a PhD in neuro-oncology from the University of Amsterdam, UMC, and has focused on combination therapies for glioblastoma. She is also a registered toxicologist in the Netherlands with Eurotox, And since for the last 2 years, now 3 nearly, has been working on Medstone science using AI to translate clinical and toxicological data and to foster more consistent decision making. So at Medstone Science, she applies Cirrus, which is the company's domain-specific NLP model, to categorize and structure scientific literature with LLMs daily. So to develop more evidence synthesis across therapeutic areas. Megan today will be talking to us actually about this, how through her work, she is enabling smarter drug development through clinical data interpretation through the use of large language models. Megan is here live with us, so please don't hesitate if you have any questions for Megan. We have plenty of time for questions after the end. Megan, over to you.
Beste mensen, graag uw attentie. Bij een stroomstoring en onze noodaggregaten zijn aangeslagen. Probeert u alsjeblieft zo min mogelijk de licht.
You have a colleague who's talking, I hear. In the same room.
Ladies and gentlemen.
No, not an emergency. I will wait 2 seconds.
Okay, so if you could mute yourself and let us know. Here she's starting to share her screen. And once the background noise has gone, I'm sure she'll be able to start her presentation. If you could put it in presentation mode, please, Megan.
Yeah. Do you have it now in presentation mode? Sorry, guys, for the
No,
loud
it's in, we can see your next slide, you know what I mean. It's in the presenter mode, not presentation mode.
Let's see, sorry.
It's okay. Okay, the background noise has gone now, so.
Yeah, sorry, there wasn't a, they were saying something about emergency energy aggregate that was not working. So
okay,
to use the
so
elevators.
Hope nothing will impair your talk.
No, I can still do my presentation, so that's good. But I will see the sharing. We tried it this week, but I don't know why it's now, because it should be content only. But then I'm also checking with the. Maybe that's why. Okay, wait.
Presenter mode.
Megan, if you go to the top on display settings.
Yes, second tab on the top. So where you were, Megan?
2 seconds. I think it had to do with my double screen, so that's why I disconnected it. Because that's something I didn't have this morning, and that's why it's different. Let's see if it's now working.
It's not in presentation mode yet. Perfect. You can start. Over to you, Megan.
Yeah, thank you for having me and for being patient with me getting this sorted. So today I will be talking about enabling smarter drug development through clinical data interpretation by LLMs. And I was already introduced by Janet, so thank you for that, Janet, for the nice introduction. I have a background as biomedical scientist, indeed, with a PhD more on the practical side. So I did a lot of research into glioblastoma and therapy efficacy, but after that switched more to the computer side. And since a couple of months, I'm even managing director at Medstone Science, where we use NLP and large language models mainly to enable companies, even academia, to interpret clinical data. And I will take you through some of those slides today. I will be speaking on personal capacity today, and there are no conflicts of interest to declare from my side. So during this talk, I firstly will start with clinical data interpretation, then what interpretation really means in this case, then how you go from traditional natural language processing models to large language models, the application in the toxicology and pharmacology in this case, risk and limitations, and lastly, how large language models can be used for clinical use. And the goal at the end of my talk is that I want to build awareness of what to consider when using large language models in clinical and toxicological context, because I think that is, we cannot refuse the use of large language models, but I think it's really important, and what I've seen during my daily work as well, is that people understand how to use them, the limitations, and if you know those, then you can work really nicely with them and optimize your clinical data interpretation. So that's already a little bit of a giveaway what you can expect today, but let's start at the clinical data interpretation and the current challenges. Because the clinical data, large evidence or large fraction of this evidence is unstructured text. And what do I mean with that? You have the clinical study reports, which have free text sections, individual patient events, clinical relevant safety and efficacy details, but this is all written in reports, always in different structures. And although it has sections in it, every researcher that fills in these reports writes it differently. And that is affecting, of course, how the clinical data is structured. Then you have adverse event descriptions. As many of you maybe already know, they can be described differently, categorized differently, different context, timing, severity, causality. All of these things are important in understanding what the adverse event text means and giving it more value to it. And the same with medical reviews. You have expert clinical or toxicological assessments that give interpretation of these events, patterns that happened. You can only see that when you have more bulk of data. But if you do this manually, it has a high cognitive load and a low scalability. So that's why we're also looking at LLMs or natural language processing models to scale this. But what we see is that interpretation is the real bottleneck. And what do I mean with interpretation? I want to take you through an example. First of all, it's important to understand what entity extraction is. And by interpretation, you want to do more than entity extraction only. And I will show you in the next slides. For example, here we have an abstract. We don't have to go into detail of the abstract, but entities that you could get from this abstract is, for example, an entity is 600 milligram or hematological or manageable toxicity. These are all fractions of text that can be extracted from this abstract. And if you then think about the extraction, the context matters, and that is point 2 in this case. What do we mean with interpretation is that the context is important. So you need more than just that small fraction of text. You often need the text around it, even maybe more other relationships that are explained in the abstract. In this case, if I need to give an example of how to understand the text, in this case here you see, for example, written arm 2 closed early secondary to toxicity. If you only have this sentence, you don't know what arm 2 is. So if you would only have the entity arm 2, you will not understand what arm 2 is because it's explained in different sentences before. So you need always the context of the information. And third, you need to connect the evidence to decisions. So interpretation that links observation to clinical reasoning that support your safety and development. And in this case, for example, the same abstract, you see here that the first sentence that I, for example, took here, arm 2, closed early secondary to toxicity. And then you see the sentence afterwards is saying 21 patients were evaluable for dose-limiting toxicity. That is the explanation that comes along with this first sentence. So all these kind of details are important that we as human beings are able to integrate. But if you want to use AI in this case, you need to be aware of what AI can do and what not. So lastly, and that's point 4, is that the handling of the ambiguity and uncertainty is also important, and clinical data is often incomplete or subjective, or there are interpretations of the data. For example, in this abstract, it's written that it has the combination that was tested here, for example, has a manageable toxicity profile. What do we describe as a manageable toxicity profile? That's not defined. It's not defined by us as researchers as a one thing, but it is words that we use. And this is seen as a data point because it's text in clinical data. But it's really important to know that this is, for example, for an automation, hard to take along. But I do have to say, over the past years, something interesting happened, and that is that you have the traditional solution to automate this nowadays is with natural language processing. These are models, and I'm not sure if someone talked about it today because I was unfortunately not able to join the talks in the morning and the early afternoon. But what we saw is that there was a transition from manual to automated text extraction, and this was enabled by natural language processing approaches. This is also something, for example, that we use at our company, and we saw the early adapters were there in 2000s, but the main improvement came when natural language processings were involved, and it was between 2013 and 2017. And if you look at what natural language processing models can do, I summed up the pros and cons of these models is that they scale beyond the manual review. That's also why they are developed. They have quite a high consistency. They can speed the insight of the generation of insights compared to manual use. They structure unstructured data. So what I mean with if you have this abstract, it's unstructured. Every abstract is written in a different way. Of course, as a scientist, we have a certain format, but it's never written always in the same that you find, for example, those always in the same number of sentences. It can be really different, and that's what NLPs are able to do for us. And thereby they help us to make expert decisions, or they help us as experts to make decisions. What we do have to see is that their limitations is that their NLPs are rule-based often, and it is also a downside because that means that you need to explain to an NLP model what the rules are. So you need to develop them, you need to maintain them, and that's also why it's costly to maintain them. And you train them for a specific indication. So that's point 3 on this side. And therefore, it can be difficult to transfer it across different studies. And there is limited semantic reasoning. So by knowing these downsides of NLP, we luckily had something new over the past years, and that are the large language models, where my main talk is, the main topic of my talk. And this is really, as we all know, the first ones were there in 2019, the generative large language models, and they demonstrated a broad language capability. And in 2022, and from then onwards, they were adapted. Sorry, I have been a bit sick, so my voice is not helping me today. But they have a widespread adoption with instruction-tuned and conversational models. And here again, I want to pinpoint or show you the pros and cons of LLMs in general. So they have a strong semantic understanding. They handle language variation naturally. So for example, if you have abstracts are not always written, for example, in English language. So it can sometimes still be in Chinese, or we often even see in our work sometimes French abstracts. That's where LLMs can help really well because they automatically can translate it and still give you the same results. They can also help with cross-document reasoning. So if you have different kind of documents, so in NLPs, you always want the same kind of input data. So for example, in our case at the company, we always use abstracts. But with an LLM, you can say, okay, one part of data is coming from an abstract and one other part is coming from a clinical study report, for example. And then it's still able to give a summarized result from both of them. And that's also the next point, the few-shot adaptability. And it also is able to capture implicit context and relationships. So you can ask it, okay, I want to know something about a dose. Can you give me back in which for which drug it's given and in combination with which drug. For example, if you have multiple drug or multi-drug combination studies, then this kind of information is really helpful. Although we also know that there are downsides, of course, of large language models. And I think what has been the main talk over the past years is the risk of hallucination and not inherently reliable or factual. So it is able to make up stuff itself. So if you don't prompt it right, and I think many of us have experienced this already, maybe a couple of times when you're a frequent user of the generative AI or ChatGPT or Copilot or those kind of LLMs, then you see this happening. And when you don't structure it right, you get hallucinations. What we found really a downside in our line of work is that there's a weak traceability to source text. So sometimes, even though you give it a restriction of only giving back scientific sources, it links scientific sources to the wrong sentences. So that is really something that's a downside of the use of LLMs. The uncertainty calibration, it still has, even with the newest models, it still has difficulty to do that. And you really need to give it safeguards for regulated use. So if you want to really use it in practice, you always still need to have a human in the loop. And that can still require a lot of time. So LLMs overall have a semantic power, but require constraints to be trusted. And I think that is really important to keep in mind. But if we look at the pros of LLMs, we believe as well, and that's also, we have seen it, or I have seen it in practice, that it can help us as scientists to more efficiently and faster interpret our clinical data. And I want to show you a couple of examples how it's applied in the toxicology. And I think it's more broader. It's not only toxicology, but pharma toxicology combined. For example, I summarize here a couple of articles which have really used it in an interesting way from my perspective. Here, for example, the first one, a large clinical language model to unlock patient information from unstructured electronic health records. Or here, this one was focusing on adverse drug events. Overall, you can say that there are definitely examples where recently peer-reviewed articles that are illustrating the application of large language model in toxicological analysis and interpretation. So it can be successful, but you need to know how to use it. So I believe that, especially in the pharmacology and toxicology field, is a good test case because we have such well-constrained way that we want to receive information. It's primarily text-based. You can, here I say causality often implicit, I think depends on case, but there are strong cases where you can make it, or there are cases where this is quite strong. It is really dependent, of course, on context. So there you really take the most difficult situation with toxicology and pharmacological cases. So if your model performs well or your pipeline performs well on this kind of data, that's really a benefit for if you want to use it for any other medical interpretation data. So as I say, it's a good stress test for interpretation quality. And that's also why I like to use our pipeline in this kind of cases, but I will not show any of those today. But again, what it does well in toxicology setting is that it is able to link symptoms, drugs, and timing. And what do I mean with it? For example, if there's a case where you have a paracetamol overdoses, LLM can link it to nausea, then to rising ALT and AST levels, then it asks for the time since ingestion. It can even look up the risk of acute liver failure. This is all kind of information it knows. It's known for from data, and that's really helpful. Secondly, is able to normalize heterogeneous terminology, and that is something where we have been using it quite a lot. As you can see here, you can have in written text, you can have multiple kind of words, entities in our case, that explain exactly the same. And you can ask LLM to help you with one standardized toxicity endpoint so that they all come in one categorized group and that you can better see how much data, for example, there is available with liver injury. And we call this an ontology, creating an ontology, and that therefore it has reduced a lot of our time, for example. Another one is shortness of breath, dyspnea, respiratory distress. You can categorize this also to one event, or to one entity. Then it's able to integrate information across documents. If you, as I said at the beginning, if you have multiple kind of documents, you can combine them into one safety signal. Of course, you need to give it constraints, but as long as you give the LLM proper constraints, it does really well in creating these kind of documents or even signals. And the same is for if you combine a case report with lab values and medication history. And this is something we are, for example, now setting up for a pharmacy, this kind of information. And it does take a while to set up, but as soon as you have it set up, you can really just press enter and run your LLM. And that is really, for the future, helping a lot. Then capture contextual meaning, as I said, it can distinguish this between no evidence of nephrotoxicity observed versus nephrotoxicity cannot be excluded. This kind of nuances, it is able to detect it, and especially if you prompt it in a proper way. Another example of that one. But as I said at the beginning, what I also find important and want to share with you today is also the risk and limitations, because as soon as we know the risk of limitation of using these kind of models, then you also know where you need to be alert on when using them. So I also use a couple of references here, but this review, the authors of this review also concluded that they mainly see that LLMs are used in academic research. And I can understand that. And that I believe is due to the following risk and limitation. So as I said, the hallucinated causality. Sometimes correlation are framed as a mechanism or causation in situations where that's actually not true. And if you once see this as a scientist, I can understand that you become skeptical and you don't want to use it in real life. And you only want to use it in an academic setting. And then you have the loss of uncertainty. Can, for example, say in the text it's possibly related, and then the LLM makes drug induced from it. So it collapses the data into information that is not what's actually written there. And that is something you need to be aware of. My team, for example, has been training, has had a lot of training in this and knows how to prompt your LLM, but it's something when you are starting using LLMs for this kind of applications, you need to be aware of these downsides. Reporting and annotation bias. As I said, even though we as scientists know what the structure needs to be of an abstract or even an article, we all know each journal, for example, asks for a different structure. So, and that's only structure, but you have writing style, you have country differences, for example, or even regional differences. So all this kind of information that can bias the results. Then, yeah, overconfident synthesis. That is something I find always funny to see because I notice it when it happens, but I can imagine when you're first using it and maybe not aware of the downsides, then you think, oh, it has really nicely written a summary for me and it seems like really logical. But then when you dive into the documents that you gave it, for example, you're like, that's not what I read back in there. And then, so it's really overconfident explaining, for example, that there was an effect. Although when you read back the documents, you see like, okay, the writer saying there was maybe an effect, or we saw a small effect, those kind of things. Context insensitivity. This is a bit the same, but it's more about missing the confounding. So disease severity, timing, just so more, yeah, all the information that you maybe use as a toxicologist as well to make a risk assessment. It can be lost when using LLMs. And regulatory risk, what we see it becomes a little bit lazy, let's say, if you reuse it multiple times. So for example, you send in one first batch of documents and it does it really well. But then you send in the second batch of documents and you ask exactly the same question, then you see that it's not coming back with the same quality. And that's what I mean with lazy. You need to be careful with how to apply it in that case, because there are ways around it, but it's difficult still. So knowing this all, and I think one of these references that I cite here, they explain it really nicely, and that's why I just kind of used it from them. They say to proper research and practical implementation of LLMs in an effective, safe, and responsible manner, all the above risk and limitations must be understood by many people from diverse backgrounds. And with that saying, I learned over the past 3 years, for example, a lot from the more technical people in my team, because I didn't have this technical background and I wasn't aware of these limitations. But since I am, I can help other scientists to understand them as well. And as soon as you understand them, I think you're more safely using LLMs. And there will still always be stuff that we don't know. But if you already start with these 6, for example, or even with one of them being aware of it, already helps. But I think this is still maybe the most difficult approach in using generative AI in general, and in this case, large language models, is that people come from such diverse backgrounds that it's really hard to get everyone on the same level of knowledge. But saying that, I do see new developments in the field that make me enthusiastic about the use of LLMs and also covering these limitations, because one of the newest developments or implication over, I think, the last couple, maybe 1.5 to 2 years, is RAG. RAG is a retrieval augmented generation. And what does RAG do? It helps large language models to constrain large language models. So when you sometimes, as I say in the risk, in just a slide before, what you see sometimes is that they just go a bit from left to right and they don't constrain to the assignment you gave them, or they start to make the summary nicer than the actual picture is. The RAG tool can help with that. And here I cited 2 articles that also used it in clinical setting. In this case, for example, they call it grounding of these large language model in clinical evidence. And it's a RAG generation system which was used on the UK NICE clinical guidelines. But what does a RAG do? I wrote here quite an intense text, but so what it does is a RAG makes kind of a binary code of information that came from the LLM. So it runs, we call the user query and information in a vector database. And that's what I mean with binary code. Based on that, it's restricted to come back with that retrieved information, you then query your large language model again, and then give back the results you want. It's really technical, and if you want to know more about it, I can explain more about it and even couple you to one of my technical team members who can explain it way better. But it's something that we have been using and really helped us also to constrain the LLMs and use it more safely, also by people that don't always are aware of all the risks and limitations. Lastly, another example that I wanted to show you is this clinical guidance tool. That's where they used an LLM, and to be specific, this version of an LLM to extract relevant information from the UCLA guidelines. And after they did that, they asked 7 doctors from the ward to assess the chatbot performance by comparing its answer to the gold standard, evaluating it on relevance, completeness, and measuring the time taken to find answers. Because in the end, we are using LLMs to speed up the process and to automate stuff. So what they saw in the results, for example, here in this case, is that 73% of its responses rated as very relevant. It had 100% recall for the extracted guideline. Oh, so it's double guidelines. And 78% of responses were rated satisfactory. I think that's pretty high, because if this means that you could automate 75%, approximately 75% of these processes. There's always 25% which still needs to be checked, but if that needs to be done by a human instead of 100%, that helps. And if you look at the average completion time of 10 seconds compared to 30 seconds for human respondents, I think this is a nice example of how you could use it. And then lastly, what I want to take you, the last couple of minutes, I don't know how I'm in time, but I want to take you to the design principles, because as I said, there are a couple of things that you should take into account according to my own beliefs in when you're designing a pipeline with large language model for clinical use. So human in the loop by default. So I think that's really important. You always need an expert to review it, especially if it's clinical data that you are assessing. And this evidence first output. So yeah, it needs to link to every source. It always needs to link to something. So there is no free text conclusions. And uncertainty preservation, again, hedge terms, alternative explanation, and missing data are explicit surfaced. Context anchoring, prompting and pipelines, enforced drug indication, population dose, and timing constraints to avoid the decontextualization synthesis. Governance and versioning, you need to be aware of which version you're using, what your prompt is, which data snapshots you're using. And important here for what I believe is that you need to design a system to feel safely visible and reviewable, because when you see when it fails, then you know and you can act on it. Janet, am I still okay to do like 2 to 3 minute slides?
A couple of minutes, if you don't mind. If you could conclude, that would be great.
Yeah, then I will make it, that we can do that. So I made like a practical flow of ideal situation. So as I said, the clinical data that I'm speaking about is textual data. So if you have textual data from articles, any kind of document whatsoever, as long as it's text, my ideal situation or my ideal flow would be that you put it into an NLP, so natural language processing model, so that structures and extracts the data that you are interested in. So think about drug dose, timing, adverse events, labs, all by just these fractions of small text, because then you have it structured. From there, you put it into a rack. Then the rack can take care of the evidence grounding. So it only retrieves relevant source chunks. It can also make the constraints that you need for your LLMs. And based on that, you have a bounded evidence set So not everything goes into LLM, but really a bounded set. And as I said, then you go to your LLM, which does the interpretation and synthesis of your results, which in the end, you end up with, review-ready insights. And I have to say, it will never be the regulatory truth, because that's why I said there's still a human in the loop that does the last check on the data. So that needs to review and give it even maybe feedback to optimize even your pipeline. And then, so as I said, the human in the loop is the control mechanism. And I think if we are all still aware of this fact that it's needed, then you can safely use it and use it for smarter drug development. I think here I covered this all. Yeah, so smarter drug development comes from better clinical data interpretation, and I think that can be done by large language model as long as it's governed by experts. So my take-home message for today is that the value of LLMs lies in augmentation, not automation or replacement, because that's what we have noticed in practice, that people are afraid of, that it will replace you. And I believe we should never make that happen. It can only help you to augmentate the data and make thereby expert decisions. And it does enhance speed, consistency, pattern detection, because it does that sometimes way better than we can. But as I said, retain the judgment, responsibility, and final decisions to humans. I think that's what I've seen in practice, really an important take-home message for me. And that was it for today. So I'm happy to take any questions, but I think we maybe also go to the panel discussion. We'll leave it to Janet.
Yes. So thank you very much. Again, I think that was extremely clear, extremely practical, and you really showed, I think, how things are done in practice, which is something, we all hear about AI. We're not technical persons. We're not trained. This is something we're learning as we go along. And things are moving so fast that we don't only provide, like at my university, don't only provide training for the students, but actually training for ourselves as lecturers who have to manage sometimes the students more than us about AI and how they use AI, especially for their assignment. So this is something really practical. We already have a first question here from Amia in the Q&A, and I invite anyone else who may have a question to do so. So the question is this: do you find, or have you tried to see whether error scaling up when reused across assessments are reduced by using reactive AI, that is, only using current inputs without retaining memory?
I think that Amaya, thank you for asking your question. I think what you're referring to is when I said, like, you see that it becomes lazy when you put in prompts multiple times. I have to say, I don't know exactly what my team does, but I know they were able to get out of this loop. I should ask them probably how they have done it. But yeah, I know it's feasible, but how exactly, I don't know. But what you often see is and then I take myself as an example, when I use the front end, I would say, from these models. So just, hey, you go to your internet browser and you use the LLMs. We forget that this kind of things happen. When you have programmers in your company or people that are more technical skilled, they definitely know to get around these kind of hiccups. But it was mainly focused on people that are using it from the front end kind of way.
Okay. Thank you. We have another question. I think, I'm not sure if it's for Megan, so I assume it's for you. It's somebody who didn't write their name. So CSR development is guided by CHE3, MedDRA, ICDs, and consort structures, and they're also part of the regulatory and public discourse documents in clinical development. When you say that the CSR reports are written differently by different developer writers, can you further elucidate the issues?
Just for me to understand, with elucidate, the meaning is of that to what I can further explain.
Describe the issues, what happens with CSR reports in in putting them in these models?
Yeah, so if you look at CSR reports, there's all kind of information, of course, in there. Although I have to say, because our NLP model is trained on those kind of documents, because it's really structured and it has, of course, the sections. But what you would see is that in one study, there is written, okay, the adverse events were seen with this doses, these amount, the number of patients. But you will see that not in each CSR, it's written always the same amount of information. So some will be way, let's say, way more dense than the other ones. And then you are missing stuff. So there you already get your bias. And you could only prevent this by saying that you need to almost, I think, run an experiment yourself, is that you say, okay, doctors, we are gonna, because it's often written, the CSRs by doctors, you are part of a research and you need to always fill in this and this and this information. Even though we are doing, then you still see differences in the outcomes. So I think that always stays a problem, but you can train your LLMs on it and at least specify which information you need. And if it's not there, that's what we always do in practice. We want the LLM to also specify when it's not there. Because if you don't do that, if you don't say specify when it's not there, then it starts to hallucinate. Because it feels it needs to come with an answer.
Okay.
I hope that helps.
Yeah, we have some other questions in the chat. So this is from Hanjian. How do you track, validate performance in terms of accuracy of the results retrieved? Are you referring to commercial LLM models where underlying parameters can change? If so, how do you validate, track performance of models over time? There's a lot of questions in one. Do you prevent transfer of proprietary sensitive data to commercial parties like LLM providers? Do you automatically check NLP analysis of text with data and tables to verify if numbers correlate with the text? There are 3 questions here in one. So I start with the
there are
first
many.
one.
I'm thinking where to start.
4 questions. How do you track, validate performance in terms of accuracy of the results retrieved?
Yeah. So for our NLP, that was easier. We recently also published our model, and it had an accuracy now, I think, in the publication of 95%. But in the end, it's also that our model is reviewed, of course, by experts in the field. So in the end, it is based on what you know, and we have their training and validation sets and those kind of things. With LLMs, indeed, it's way more difficult. So then the validation and accuracy is based on, and is tracked based on the human expert in this case. And we have kind of a rule there that we create a panel of human experts. So it's not one person. So, for example, for a client of us, we're running quite already for a long time a project where we use LLMs and our NLP model. There we always say 3 experts. And it's kind of the ruling with the same as with experiments. If then 2 say yes and 1 says no, or like that kind of, and it's an open discussion. And we need to be aware of the bias that we there have as human beings as well.
Okay.
So I think that's to the one.
So the next question is, are you referring to commercial LLM models where underlying parameters can change? If so, how do you validate, track performance of models over time?
Yeah, I think this question is related to that the commercial LLM models, of course, have all versions. And yeah, you need to keep track of that. We are using those as well. The underlying parameters you need to be aware of. That's something, for example, that my IT team tracks. And as soon as you change it, then you also need to validate again and track your performance. Yeah, definitely true.
All right. Now, how do you prevent transfer of proprietary sensitive data to commercial parties like LLM providers?
Yeah, that has been privacy regulation in this part is, of course, something you need to be aware of. And I think European-wise, I've been in contact with someone that was also involved in creating the European AI Act. It's something that we slowly start to get rules about it and how to use it. In our case, what we always do, and I think that was also on one of my slides, that you need to know when it safely fails, like that you need to know when it fails. And those kind of things. So what we do is open communication to our clients. Hey, we're using this commercial model. We can also use other commercial models, or we can even run it on our own servers. Then it's our responsibility to safely use it. So that is always one of the last options, but it's also a way more expensive option, because that means you need a lot of server power, let's say. And my colleague was just saying today, like, yeah, we can all run that ourselves, but then we need a whole ocean to fuel the whole system, because it requires a lot. And that's about the commercial LLMs, more easy because they have already that infrastructure.
And the last question from Marjan was, do you automatically check if the NLP analysis of the text with the data and the tables to verify if the numbers correlate with the text?
Yeah, I have to say NLP and tables is not a good match in our case. So for that, we are still always referring to our clients like that's manual work, to be honest. So yeah, text extraction we can do. Table extraction, we are becoming with the NLP models and the other large language models and what we're doing worldwide, we're becoming better in it. But text is, and especially, for example, figures. For example, if you want area under the curves from a really nice figure, that's still difficult to do.
Okay. Now, another question, we don't know from who. How do you think in silico trial design can help rare diseases clinical development, especially when there are insufficiencies of data in rare disease?
Okay, I think this is partly to referring to the first presentation of this session, right? The in silico trial design. Let me see. Yeah, when you have insufficiencies of data, that's difficult, and especially in rare diseases, because, for example, I have been studying glioblastoma, which is also a rare disease. There's not a lot of data. We have been, at that time, we were already trying to use not NLP models, but yeah, we have been using NLP models as well a little bit. But for the in silico trial, I need to, I haven't heard, or I did listen in, of course, today, but I don't have the expertise, especially on how they do it. But yeah, when your data is scarce, then it's difficult. I agree. And then you have to do it with what you can. So then in silico trial design, yeah, if that's your next best, I would definitely use it.
Okay. So if there are any questions, please, now is the time in the chat before I hand over for the concluding session. I do have a question myself, if I may. Yeah. Which are the major barriers do you see yourself on a practical basis every day? How long does it take you once you have a query?
Yeah.
How long does it take you to come out, to go through that flow chart you showed us, to go through all the checks and you're happy with the results? How long does that take? Or does it vary on the query being made?
It really depends on the query made. I have to say, for example, today I was discussing here with a couple of new colleagues. For example, our NLP model, if we, I was explaining what we do there is, for example, this week, one of my colleagues started with 50,000 articles of interest and reduced it with our NLP in half day to 150.
Gosh.
But then from those 150 and the steps in between, the filters that you use, you need to validate it. So that's what she did the rest of the half her day, let's say.
Mm.
So then you have one day going from 50,000 to 150. Then those 150, you want to give that to your rec with all the information that's there. Because if you can imagine, you have your abstract. How many entities are coming from one abstract? An abstract is, I don't know, how many words are there always that we are restricted to? I don't write them that often anymore.
300, 400 abstracts.
Yeah. So let's say that each word is one entity. And then multiply it with 150. That's the amount of entities that you get from. Then you're thinking, oh, I only have 150 abstracts. But that's multiplied with the amount of words in there is quite a lot. Then you feed that to your rec, and that rec needs to be developed. So that's not a day. That costs a little bit longer. But we always say to our clients how you can imagine it if you make it is the investment, yes, takes time. But then as soon as the pipeline is there, then you can repeat it every day. You can just press enter and repeat every day as soon as you have validated it. So the validation takes time for certain projects. I would say roughly 2 to 3 months it will take us to validate it. But then you have and then I'm talking about quite a big project, let's say. Then it takes 2 to 3 months to validate it. And then from there on, you can just repeat it. And we see with our clients that it saves a lot of money and that their human resources can be used for better use. And that's where we are going with.
Okay.
So not to replace people, but really so that we as human beings can focus on stuff where we are actually needed.
Thank you. I'm not sure if there are any queries from my colleagues or any more queries from the participants. Thank you very much, Megan, for really answering all those questions in a lot of detail. It's really something which I'm sure if we had to repeat your presentation next year, I'm sure things would have moved so fast.
Always happy to give it once more. No problem.
We look forward to meeting you again anyway.
Yeah, thank you.
Thank you very much, Megan. That was really, really excellent. And I'll hand over now to Ian to conclude for closing remarks and as well, very important for the prize giving. Ian.
Great. Okay, thanks a lot, Janet. And thanks to Megan for her presentation there. So, we're approaching the end of the meeting now. Before we close, I wanted to announce the prize winners from our oral communications session. So the presentations were ranked by 3 independent judges, and they took into account the career stages of the presenters as well. So the winner of the oral communication prize is Mark Ellison. So congratulations, Mark. You'll be receiving £200 in cash. And the runner-up is Zhonghui Hang. So well done, Zhonghui. You'll be receiving £100 in cash. So we'll be in touch after the meeting to arrange payment of your prizes. But congratulations from all of us on the organising committee. So to wrap up then, on behalf of everyone involved in the organisation of the meeting, I wanted to say a big thank you to all of our speakers and all of our attendees for participating today and making this such an interesting discussion. I think we've learned a lot about the opportunities and challenges associated with the increasing use of AI throughout the different stages of developing a new medicine. And I know this is a wider topic of discussion within both societies, within the BPS and the BTS. So once again, please consider at least attending some of the events that we're hosting over the coming months. And also consider becoming a member if you're not already a member of the societies. And then you can interact with us and help frame the direction of travel for the field over the coming years. So that's all I wanted to say. That brings us to a close. Thank you again to everyone for your participation. And we look forward to seeing you at future BPS and BTS events. So thanks and goodbye. Thank you.